we have announced this workshop a couple of months ago, and we are very happy to tell you that we have over 250 people interested in participating and registering to the meeting, covering 50 countries, and on most of them scientists or students, but also more than 35 breeders. So it's good that we can also communicate with breeders what we know in academia. So without any further delay, I'm going to start with the first talk. This in general, this training school is about genetic resources and linking genotypes to phenotypes and the other way around. So finding the basis. Maybe Simo, you can mute everybody else.
Yes.
Thank you.
We'll do it.
So the first part of the program in this morning session. Can you do it?
Yes.
Okay.
You have to find who is.
Okay.
Okay.
Everybody else, please mute your phones or microphones. So the first talk is by Björn Usadel, who's gonna talk us about the genome of Solanum pennellii. That is, as you know, this accession has been very useful for mapping and finding the genetic basis for many traits that are important in tomato. And so thanks, Jordan. The floor is yours.
And let's see. Do you see this in full mode? Okay, so thanks for inviting me here. I'm talking about the Solanum pennellii genome, and I'm going back a bit in history and tell you a little bit what it is all about. So the Solanum pennellii is a very remarkable species. As you already heard in the introduction, it has been used for many traits, and that's because it comes from a more arid region, and this is very drought tolerant, also salt tolerant. It's tolerant against multiple abiotic stresses, and that has been used in multiple experiments. So I just show some of them here, and you see that actually as back as 1977, people actually looked at that and saw that. And if you just look at this, here's some simple water stress experiment where you withhold water to Solanum lycopersicum, the cultivated tomato, or Solanum pennellii. You see there's a very, very stark difference in its way to cope with drought stress and, I say, withholding water. So there's many papers describing this. And mostly when we talk about Solanum pennellii, we talk about a famous accession, LA716. I'm going to come to this in a couple of minutes. Just to give you some background why this is interesting for us, and I'm just reproducing the slide of the Moyle lab here, that Solanum pennellii is down here in this section lycopersicum. So it's very, very far removed from our cultivated tomatoes and our red-fruited tomato. It's a green-fruited tomato, but despite it being relatively far away and maybe like more than 2 million years of splitting point, the point is you can still cross Solanum pennellii to Solanum lycopersicum. I stole this slide here from Itai Offner's paper and Dani Zamir, who will talk, I think, about the bill lines later. And because you can cross Solanum pennellii, and that has been done in the Zamir lab extensively, to the cultivated tomato, you can actually try mapping many of these advantageous traits from Solanum pennellii by looking in the offspring population of Solanum pennellii and Solanum lycopersicum. So it's really a fantastic genetic resource. It's an exotic germplasm that you can use, and it contains many, many traits and many QTLs. A few thousand QTLs have been mapped already using this resource. So the question is, what does make Solanum pennellii so stress tolerant? So because we saw this, of course, it comes from such a region, but of course we're interested in finding the underlying causes for the phenotypes. And because we could see that this is a genetically tractable thing, so if you can map QTLs, it's genetic. And of course, then you need to understand what the genomic basis of this is. So actually, almost 10 years ago, when I was still in the Max Planck with Alistair Fernie, we set out to sequence the genome of Solanum pennellii, and this is one greenhouse, and we actually needed to harvest tons of these plants to sequence that at that point with short read sequencing. That was successful. It took us more than a year, together also with the help of Tübingen, Weigel's lab, and others. And we got the first genome version of this LA716 that had been crossed by Dani Zamir earlier into this Solanum lycopersicum. So did this help? Well, Jos Rose at that time, he looked with us at the cuticular and wax components, and what you actually see is, if you just look at gene expression, you see the cutin, some wax genes, and some cuticular aromatic compounds, both inside Solanum lycopersicum, which is always going to be red, and Solanum pennellii, which is blue-green. So you can see the difference, and you see that actually most of these genes are much higher expressed in Solanum and pennellii, where some wax components are showing the opposite behavior. And that is actually more or less in line with what you expect and feel, and you can actually look at the cuticle. It's different. If you touch pennellii, it's very sticky, which is another compound that Solanum pennellii makes. So that might be an explanation, because of course, if you make more waxy components and more cutin, you can protect the plants against water loss. Surprisingly, that was not all, because when we looked at the transposons with Florian Maumus from INRA AFSI, and he, of course, Solanum pennellii has a slightly bigger genome than Solanum lycopersicum. This is partially due to transposons, but you see it's mostly quite a lot of difference in gypsy transposons and maybe a little bit of more copia type transposons. If you don't know what these funny names mean, these are just different transposon families that you typically find in plant genomes. There's a lot of other things which you find. It's not necessarily transposons, it's other repetitive elements like viruses. But I just want to point your attention to these two. And the interesting thing was when Felix and actually looked, Florian, sorry, looked at the transposons and looked in Solanum pennellii and Solanum lycopersicum, and they did a sequence similarity, where a sequence similarity for this repetitive elements is actually showing you something about the age. If the transposons are still very, very similar, which is here on the right-hand side, that means they must be recent, because there was no mutations happening. Whereas if they're very dissimilar, more on the left-hand side, they are more ancient, they have had time to evolve. This we are looking at really, as you see, a huge number of transposon copies, up to like 20, 30,000. So what I want to point your attention to is actually that something happened here. It's just this blip where this red line goes up in Solanum pennellii, where in Solanum lycopersicum, this line goes down, saying actually that here a recent transposon burst must have happened. So we have a new copies of these transposons. Well, that is interesting, but what could that mean? And by actually manually, by somebody in Alistair Fernie's labs, he just looked and said, well, it looks like all these transposons are always sitting next to genes which I know are involved in stress responses. And when we did some statistical analysis, this was indeed the case. So in the upper row, what you see is the insertion, if you just randomly assign genes and associate genes to transposons, And you see, well, of course, this is more or less the same, no matter where you look, this is always distance of the transposon to the gene. And here, of course, the same with the random assignment. Whereas if you just look at these copia transposons, now to stress genes, so genes involved in drought stress, heat stress, you see that they are actually much more likely to insert quite close to genes. Whereas in Solanum lycopersicum, you actually see the opposite behavior, that if anything, they're de-enriched in this region. So there might have been some selection or some fortuitous coupling of certain transposons to genes involved in stress, maybe driving or changing their expression. This is not unheard of. This was in 2014 and has been shown several times, of course, because natural evolution takes place. So that might explain things. However, the point was, of course, this is one pennellii. And let me just show you this lost pennellii, as Dani Zamir has christened it, which was found in a collection of the IPK Gartesleben by Dani Zamir. And this is, of course, a completely fake picture. And this was also quite interesting. And at that point, we once again had the question, can we use the genomic basis of that? And at that time, that was in 2016-17, this new technology, Oxford Nanopore, came up, which we had just been using. And so we sequenced this whole genome using different assemblers at that time, so that actually put these long reads together. At that point, you still needed short read data to correct errors, and you needed multiple correction errors to get rid of the insertion and deletions and mismatches. But you could assemble a genome that was not chromosome complete, but one can use, of course, the existing resources. What I want to point your attention to is that what we get is a contigent 50 of 2.5 megabases. So what does that mean? That meant that actually all these assemblies were in contiguous sequences without any ends, where half of all the sequences was in stretches of 2.5 megabases or more. That was actually better than the previous pennellii assembly because it was based on short reads and it was riddled with N insertions in there. Nevertheless, I mean, we also looked a little bit at the epigenetics of this line, but the reviewers didn't like it, so we took it out. It's probably still in the preprint version. But basically, that was much faster, and as it turned out, that was the first higher plant genome that was ever sequenced by Oxford Nanopore long-read technology. We even bet the people working on Arabidopsis here. Now, this was again a couple years ago. So together with the laboratory of Charlie Underwood from the MPI in Cologne, we actually set out this is not pennellii. This is quite an important distinction. So to look at novel ways to assemble genomes, and they used PacBio Hi-Fi long-read sequencing, which you might know is maybe 20 kilobase reads, which are very highly precise, whereas nanopore technology gives you 40, 50, 60 kilobase reads, if you're lucky, but they're not so very precise. At that point, we still had an error rate of like 1 in every 20 to 30 bases, even though this is only a couple of years ago. And we managed to just co-assemble this genome using these different technologies and merging that. And actually, if you compare that to the published Solanum lycopersicum genome, basically you get a 1-to-1 thing. So basically, it was possible without any additional data to assemble this genome completely to chromosome level. Yeah, this is not possible for pennellii yet, because pennellii is riddled with all these new transposons, which make the assembly a bit more difficult, but you get very, very close for pennellii as well if you combine these two technologies, which, since we have published this, is actually also available in multiple tools. You don't need to use our pipeline anymore, which we developed at that time. It was actually last year. So that is possible now. And if you just look at the progression over time for genomics, so in 2014, it took us, a greenhouse full, and our contigent 50 was 45 kilobases. Mind you, everything was on the chromosome. but we had to use genetic maps to bring this to the chromosomes, and lots of jumping libraries, which might, as we now know, of course, have made small mistakes and inversions. Then in 2017, we used nanopore sequencing and get into a contigent 50 or bigger than 2 megabases. And now we get close to full chromosomes by combining different technologies for simple genomes. However, because we are interested in stress as well. And now there is not only this LA716 and this lost Solanum pennellii, which is actually remarkable, similar to accession in the RIC center. And basically, it is probably a descendant or very much related to this one in the RIC center, which is LA2963. A former group leader, who's in RWTH Aachen, she actually looked in all these accessions from the RIC center, and you see they are collected from different areas, from north to south gradient, and some of them are actually self-compatible, some others are not. LA716 is self-compatible, so is this lost one. And some of these are more salt tolerant if you just germinate these accessions on salty media with 150 millimolar sodium chloride, or you just germinate them on MS media. And what you see is this dark line is if you just germinate them normally, and you see 100% germination after a few days. If you germinate them on higher salt, after 5 days, LA716 manages to germinate, but LA1367 actually has a similar behavior if you germinate on MS media, but if you put it on sodium chloride, you see it hardly germinates. Similarly, for this lost one, it takes a bit longer to fully germinate on sodium chloride, light line here, whereas this 27119 does not germinate at all after 7 days. So basically, even in the pennellii, in the succession, you see quite a lot of natural variation between these different accessions. So we asked ourselves if we can learn something from this, and for that, together with the lab of Alistair Fernie, this is Felix from his lab, and in my lab it's Sarah. We work on actually sequencing all these accessions of the TGRC. So we have 47 accessions, and Felix wrote them under controlled salt and drought stress, and we have sequenced their genome. Felix did differential metabolite profiling, and we did some Oxford nanopore long reads, and we have analyzed the expression data, which Sarah will present in a couple of weeks in Montreal about her work in expression there. So not very unsurprisingly, if you go for an accession like LA716, this is nanopore data only. This is very homozygous. You see here one clear peak in the reads, and now you get an F50 of 7.8 just by Oxford nanopore without doing much else. Remember that a couple of years ago, with the same technology, we only came to 2.5 megabases, and this is just using nanopore long reads. Whereas if you go to the more heterozygous one, and you see from right to left, you get more and more heterozygous, then these assembly values get worse and worse, whereas the duplication becomes more. So what we actually see is, even though we don't want it, we get partially haplotype resolved genome assemblies in the self-incompatible ones because the rate of SNPs is relatively high. We have, of course, since then just suppressed that and made collapsed assemblies, which are all in this region, and which we are currently improving even further. So then the question is, now if we have that, and we, of course, want to compare phenotypes to genotypes, the question is, what do all these genomes help us? And since we actually quite active in genome analysis, and we keep a website of all sequenced plant genomes. There's more than 1,000 now available. We actually were wondering what can we do, and for that, because my lab is actually mostly bioinformatics lab, and together with a junior group leader from C+, which is the Excellence Cluster for Plant Science in Germany, she has actually developed a tool whereby you just take this row assemblies and it uses artificial intelligence to find gene models automatically in your genomes. So after assembly, which is now a relatively straightforward and easy task, it really depends on your molecular biology for most plant species. But then you get an assembly and you don't know what's what, and then you can actually get these gene models, and that's also publicly available. And what we have been doing for a long time in my lab is actually working on functional annotation. And so now we have the gene models, but we don't know yet what they do. And for that, we have another tool which is specifically only made for plants, where we manually annotate plant families in that share the same function across all land plants. Since this is a manual annotation that we do, that takes time. So we had 55–60% of all families now. The rest is automatically inferred by BLAST and similarity searches. But what it does is you seek, once again, you take these output of the genes, you submit the genes, and it tells you what these genes are, and it does that in a very reliable manner. So the basic background is hand annotation, which we have been working on for many years now. But the whole process then is completely automated for you because you don't see the manual annotation. It just has been running in the last years in the background. And so what you get is you have a genome, you run this tool Helixer from our colleague, Edan, you get the gene models, and then you get the function. So just to show you an example how this can look like, because you can also visualize that. And everything where we have these families, we get very, very precise annotations and classifications. Here for LA 716, 27119, 1734. This is, of course, what we kind of expected. You do not see differences in gene content in these accessions here in temperature sensing and signaling or drought in the drought response. So you always see the number of genes here. So you see, so for example, the central sensor protein, you always have one copy. You have, okay, here you have maybe less temperature sensor protein, but gene losses is most often something in the assembly. But generally, these numbers are quite comparable. And then one actually has to look at individual numbers to check if this is supported by additional evidence, whereas most we actually expect to be based on the analysis. Now, looking at these genes, which we have automatically classified, this is all automated now. Actually did this yesterday with Sarah because the genomes are not polished yet, but we just used them as they were. And we will look at the association to transposon genes now. Okay, if I have a couple more minutes, I mean, you could even, of course, go further out than pennellii, and that's Solanum lycopersicoides, which is actually leaving the section of lycopersicum. And again, once again, you can even cross this lycopersicoides to the cultivated tomato. And again, looking at the genome, which was a collaboration project with Cornell University, once again, Aviv Furnace Lab and Kathy Martin's lab. And that's from us seeing these tomatoes are actually turning purple, black outside. And Kathy Martin identified using this genome and comparing these genomes that we had analyzed of Solanum pennellii, lycopersicum, the potato genome and this new lycopersicoides genome, that is most likely a transcription factor causing that, whereas Cornell actually managed to show that, of course, in this respect, this lycopersicoides, and again, there was integration lines, there was differences in pathogen resistance. In this case, actually, it was partially more susceptible in certain ILs. And of course, the gene annotation helped, where Susie Strickler from Cornell actually could show that there was quite some differences going on in a particular reason for these disease-resistant genes, where two actually contain premature stop codons. So just by looking at the genes, you actually have to check again if there is something else going on, which she did and could very well show that this might be a potential cause of differences in pathogen susceptibility. So with this, I hope I have convinced you a little bit that we can use genomic resources to find what's underlying, and that we actually are providing more and more tools so that you can focus on biology. And the good thing is we get more and more tomato and Solanaceae genomes with which you can then couple phenotypes to genotypes, and you can directly look at the genes, and of course knowing candidate genes helps. And with this, I thank you for your attention. I want to thank my collaboration partners. It's the Fernie Lab, the Underwood Lab, lots of people in Granell, Sanyon Fay, Jim Giovanni, Susie Strickler, Professor Casey Martin, and the people in my lab who did most of the work were Merkel Bolger, Rainer Schwacke, and Sarah Sangisha, and we were funded by the Ministry of Education and Research, European Union, and the DFG in Germany. And we are at C+ Lab, from where we also get funding from the DFG. I thank you for your attention, and I'm happy to take any questions.
Thank you, John, for this very nice presentation, and teach us that such a huge variation, and these pipelines to identify the variation at this huge scale. If there's any question please please write it in the chat, and if you can be available at the end of the session, which is around 10:15, then we will collect the questions for each of you collected in the chat. Okay. So thanks for sticking to the time, and thank you again, John.
Thank you.
So next talk is by Shai Torgeman. Is still talking about the pennellii contribution to variation and as dissected in the bills. The talk is population structure of the interspecific LOST BILs. So please, Shai, go ahead.
Hello, everyone. I hope you hear me well, and thank you for inviting me. So today I talk on a new backcross inward lines population with new accession of pennellii that Björn mentioned it before. That is, this accession is a LA5240. Okay, so in 2007, we started to create a new bills population with this new accession of pennellii. What differentiates this accession from the LA716 is that it's fully self-compatible. It's mentioned that it creates a lot of seeds and it's doing self-pollination. So we started to cross this accession with inbred cultivated tomato, a determinate tomato that's called Leah, to create F1. This F1, we create backcross 1 back to the Leah to create 2,000 plants, and backcross 2 to the Leah. And from this stage, from backcross 2, we started to doing a self process until we reached to backcross 2 self 5. In this stage, population contained 1,500 plants. We didn't do a selection through this time, but the selection was occur naturally because some plants are fertilized. Okay, so from the F1 also between the Leah and the pennellii, we cross it to top accession. It's indeterminate tomato, big tomato for the greenhouse. So this population contains 3 alleles, and we also do in backcross 1, backcross 2, and self until we reach backcross 2, self 5. And this is the main project of my PhD. I started the project from this stage, from backcross 2, self 5. We grow all the plants in the greenhouse and create for both of this population their BIL hybrid, the Leah population. We backcross again the Leah to create their BIL hybrid, and the top population, we cross to the top to create the BIL hybrid in the top accession. So this is just a few of the variation in the BILs population. So the variation was extremely high in this population, from the leaf color and size, the inflorescence type that in the next presentation, Shai will talk on it. So you see it's the fishbone, it's fork, and much more. And the fruit characters, it's from the fruit size, from the fruit color, the fasciation, the shape of the fruit, the number of locules, and of course the melon skin phenotype that I hope that most of you know, the outside epidermis. So the variation here is very, very big, and this is what can allow us to find maps on genes. So we grow the population, the Leah population, that it's indeterminate background. We grow it on the field condition in the open field because it's determinate, and the top population in a greenhouse because it's indeterminate. So we grow the BILs, the homozygous BILs, and the hybrids of each population. And the homozygous BILs, we send them to SPET sequence analysis in Italy with collaboration with Giovanni and his lab and Paula, thanks to their help, to have genotyping of all of these BILs. So until we get the results of the SPET sequence, what we do is we design CAP markers, PCR markers, for 5 members of the self-pruning gene family and their neighbor genes. The neighbor genes, it's something about 10 to 20 kb from the self-pruning gene. And what we do, we run through all the Leah BILs population, these markers, and try to detect some recombinants between these genes, just to figure out what is the resolution of the mapping on this population. So this is the result of the recombinant that we detect in each gene. So as you see here, the number of recombinants in each gene in this population is very high. So this population, the Leah population, and also the top that I show you in the next PPT, it's very recombinogenic, and this is what allows us to find maps on genes that affect on agronomic or other traits. So we've got the data, and we start to working on the data from the SPET analysis. So this is just a few things that we've done with the data. So we filter markers that have a high percentage of NA. We filter markers that are showing high heterozygosity. Also, we filter non-polymorphic markers and non-polymorphic lines. And we filter markers with minor allele frequency that small, that 1%. And also we compare the SPET sequence to the design CAPS markers that we designed on the SP genes to show if the SPET analysis is kosher. Okay, so this is the genomic composition of the BILs of the Leah population and the top population on each chromosome. And the SPET analysis are really allow us to detect the pennellii allele in each marker and in each lines, to detect the introgression and the recombination between the introgressions that introduce through the recombination. So what we expect through our pipeline of development of this population, that each line from this population contain on average 12.5% from the pennellii genome in backcross 2 cell 5. But what we detect that in most of the genome, in most of the lines and most of the genome, we see just 6% of the pennellii genome in average in all of the population. And this is caused because segregation distortion through the pennellii allele, and it's common in biparental population with bile species. But in some genomes, as you see, for example, in chromosome 11, there are some regions that have a positive effect on the selection of the pennellii allele, and it's interesting by themselves. So what we detect from this PET analysis that in the Lea population and in the top population, that we find a recombination in the achromatic region in 17 and 18 every 17 and 18 kb respectively in the Lea and the top population in the achromatic region. So it's very high recombination rate. And in 700 and 800 kb in the heterochromatic region, the uncoded region. Okay, so after we got this, after we've got the SPET results, what we're trying to do is test driving to see if really the population is work on a mapping gene, on fine mapping. So this is a few examples of few genes that we collect in the greenhouse and in the field that I show you here, their fine mapping. So in figure A, as you see, this is the Lea BILs SP genes mapping. The SP, as you know, is the gene that affect on the growth habits of the plants, if it's indeterminate or determinate. So the Lea, it's determinate, but the pennellii, the wild species, it's indeterminate. So we collect the phenotype in the greenhouse. And you see that, and we're doing after it QTL mapping, the association between the phenotype and the genotypes that we've got from the SPET analysis. As you see, there is really strong QTL on chromosome 6, that this is where the SP gene is located. And if we look on the recombinant BILs in the figure B in this region, we can see that we can shrink this introgression to the cos genes that affect on this phenotype to 72 kb. Then this interval contain only 6 genes, that the SP gene that is Solic is here up here, the Solic in chromosome 6. It's between these 2 markers. And so it's really a high resolution for fine mapping. Also, in the Lea BILs, we collect the U genes that affect on the uniformity of the ripening, the green shoulders, as you know. So also here, there is a strong QTL that we find in chromosome 10, and in chromosome 10, this is where the known genes is located. And in figure D, it's the fine mapping of the genes, and these genes that have interval of 124 kb, that this region contain only 17 genes. So it's really a good result of fine mapping. This is just 2 examples. And this is an example of both population, of the top population and the Lea population, of fine mapping of the H locus. H is the genes that affect on the hair's growth of the stem on the stem. So the pennellii have a hairless stem, and the cultivated tomato have hairy stem. So here, both in the Lea and the top, there is a strong QTL in the same, almost in the same location in chromosome 10. And if we look on figure C on the fine mapping of the gene, this is the interval of the gene. It's interval of 210 kb, and that contain only 32 genes. So this population, it's very powerful in fine mapping of genes. It's also because it is a large. It's also because it's very big population. It contains a lot of individuals, and this is what allows us to detect genes, to do a fine mapping to some genes. Okay, so it's really hard to grow every season all this population of the top and the layer that contains almost 2,000 lines. So what we've done, it's we create a subset of line from the LEA bills, 60 lines that are fully coverage through their introgressions, the pennellii, the lost accession. So as you see here in this picture, this is the 60 lines, and each line, as you see, contain different introgression in the green one in each chromosomes. And this is what allows us to grow small amounts of plants instead of 2,000 plants, to doing our phenotypes just on this subset, to detect some QTLs that we suspect that affect on this phenotype, and after it, grow the specific recombinant bills in this region to fine map the responsible genes of this phenotype. So in this day, these subsets are in, I think, 10 labs through the world, most of them in Europe, but also Rochsar from the USDA have this subset of lines, and he grow this population in these days and doing phenotypes on it. And also Giovanni from Italy have this subset, and Toni from Spain. So this subset are we send seeds to a lot of labs that working on it. And I want to thank to all the foundations that support this research, and special thanks to Yuval Eshed from Weizmann Institute for advice and support.
Okay, thank you, Shai, for this nice presentation of the power of the bills that you constructed. So we have time, but as I said before to John, we better collect questions in the chat. So please, people who like to know more about what Shai is doing with these bills, please write your questions in the chat so we can collect the questions. And at the end of the session, before break, we will come back to the questions. Okay. Good. Thank you, Shai.
Thank you.
So we have another Shai. This time is Trigerman, and is gonna be talking about the genetic mapping of the forked inflorescence. Shai, are you there? Can you share the presentation with us?
Yes, hi. I'm sharing my screen. You see it clearly?
Yes, it's clear. Yes, thank you.
Okay, hi everyone, I'm Shai. I'm a master's student at Professor Dani Zamir's lab. In my research, I'm working on finding the gene responsible for the branching of the inflorescence in tomato, what we decided to call fork inflorescence. So first, just so you understand the term fork and what it looks like, if you're not familiar, in this slide, you can see here a regular inflorescence with one branch. In the center is the fork inflorescence with a splitting branch, kind of looks like a fork if you have a wild imagination. And finally, the compound structure that is highly branched and bears hundreds of flowers.
Shai, we have a problem. The slides are not moving. Maybe if you put it in presentation mode or something.
Oh, sorry.
They're still stuck in the first slide.
Oh, really? Okay. So just 1 second.
Debe estar compartiendo la otra pantalla. Allá está el icono de la presentación.
Maybe you are using two screens. Maybe you are sharing the wrong one. And you have maybe in the presentation mode in the other one.
I'll try it again. Just 1 second.
Try to move the slide because it looks like before.
Now, can you see it?
No, stuck with the first slide.
Oh. I'm not sure what's wrong. Maybe will get back to me later. I'll try to fix it. I don't want to waste time, everyone's time.
Oh, you can. Alternatively, you can send it to me or to Simo, which is the is hosting the session. If it's too heavy, maybe as PDF.
I don't have it on PDF.
So
תנסי בהגדרות התצוגה.
I didn't hear you well, Amir.
Let me try it one more time.
Mhm. We can see the same outline as before. Try moving to the next slide.
I'm moving the slide, but if you can't see it, I'm not sure.
No, same problem.
No, no es moving. No es moving.
And now he's doing the job. Now he's working. Good. Thank you. So the floor is yours, Shay. Go ahead.
Is it moving? Can you see it?
No, it's not.
No, now he's stuck with the first, but it moved before.
Tony, it's Dani Zamir.
Yes,
Maybe
Dani.
we can switch and I can do my gig now. I sent you the PowerPoint, and in the meantime, Shai will fix her
Okay.
problem.
Okay.
Okay.
So Saleh, because this is after the break. If you, Saleh, are there, and are you ready for presenting your talk? Hi, Saleh. So Dani, if you send yours, we'll check
I
it.
sent it to you, Tony.
To the other Tony.
Which one? Monforte.
We are in my computer, Dani, in Tony Monforte computer. So
Okay, I'll send it to Tony.
It's the same, but starting with A, Monforte. So you should have it. Waiting for the internet. Internet is lazy this morning here. Okay, lo tenemos. We got it, Dani.
We got it.
Okay.
Okay, I will share the screen and you tell me when you want to share. Okay. I'm going to change the. Okay, can you see the screen?
Yeah.
It's okay. Okay, so now Dani Zamir is going to talk about the comparative bill mapping of GQTL under deficit irrigation from two accession of Solanum pennellii. So all for you, Dani. Just tell me when you want to go to the next.
Great. Okay, so well, first of all, it's nice to be with you. And I start the talk, as always, with the TGRC slide, the Tomato Genetics Resource Center that was established by Charlie Rick and now is being handled by Roger Shetterly. And Roger is now increasing all the accessions of the tomato and the wild species and the cultivated varieties, and he's sending it to scientists around the world. He also went to South America and searched for the original accession because Charlie Rick left very precise description of the site where the collection was made. So we went back to these locations and he saw a parking lot or a supermarket or a field. So a lot of the tomato diversity was saved by the collections of Charlie Rick, and it's been used extensively by the breeding community of tomato. And if you look at the gain in yield in tomato over the years, it's increasing by 2% per year continuously. And this is much better than other crop species that did not use wild relatives. So here in the map that I copied from the TGRC, you can see the LA716 is the classical or the first accession that was collected from Atico. And today we will talk about the lost accession that comes from the vicinity in the same region. Next slide, please. Okay, so here we see the lost accession. Roger was kind enough to give it a LA number, LA5240. And if you go to the TGRC and you click, you see where the accession comes from. And in fact, it turns out you see the plant, the staked plant in the picture, and this is grown, I think, as part of the EU Soil project, and I got around 2,500 accessions from the Gattersleben collection with description, and for all the accessions but one, the description was perfect. It was exactly what I mean. I had the seed in the envelopes were exactly as described by the database in the Gattersleben, except for one accession here, which I looked at it and it looked like Solanum pennellii. And it's different from Solanum pennellii LA716. First of all, it can grow in the field. LA716 doesn't grow in the field and has other problems that I will not get into. So the idea was, let's explore the genome of this accession. And it turns out when Björn sequenced the genome, we wanted to find out where it originates from, and then Charlie left detailed description of all the seed that he sent, and it turns out that he sent to the Gattersleben location 5 accessions. So Roger grew all the 5 accessions and did some sequencing and phenotypic description, and it turns out that it's probably, the lost accession is probably LA9, whatever the number here, 2963. Okay. I selected a single plant from that accession and continued to work on it. And these guys we used in the courses. Next slide, please. Before we started to use the lost accession, we used LA716, and we crossed it to M82, which was a standard at that time. And we made 2 backcrosses of the F1 to M82, and we selfed them down till now backcross 3, or backcross 2, or backcross 3, self 11 or self 12. And so we have about 400 bills from the Solanum pennellii. They were all genotyped, and they are available at the Tomato Genetics Resource Center. So if you want these accessions and access their genotype, it's all in the TGRC. Okay, next slide. So this population, compared to the introgression lines, which were also made from LA716, that had more than 105 mapping bins, the bills had 633 mapping bins. So traits could be mapped more accurately to the tomato genome. Next slide. We were interested in the drought tolerance, and we still are interested in the drought tolerance of the Solanum pennellii. So we follow the following protocol throughout the year. When we have large populations to grow, we plant them in the bed, half a meter between each plant, and the bed is 2 meter wide. So each plant has 1 meter squared to grow in. And then we can irrigate the plants as normal. And you can see the wet treatment in the background. You cannot see the stakes. The yield is much higher than in the dry condition, where the plants get about 15% of the amount of water as the wet plant. And if you see, there's no drip irrigation because we pull out the drip so nobody opens the water by mistake. So this is a selection for in a drought tolerant, 15% of the amount of water. You can see the cracks in the soil. They go all the way to China. So if a tomato drops here, it probably hits somebody in the head or somewhere else in China. Anyway, we weigh the fruits from each of the accessions for each of the replicates. We usually have 10 replication per each genotype, sometimes more, and we analyze QTL that affect yield in drought conditions. The next slide shows that if you look at the homozygote bill, the bills that are homozygous for the introgresh segment, compared to a bill that was crossed to M82, is the bill hybrid, you can see on top that the yield, the mean yield of the bills, the homozygous bills, was 1.5 kilogram. The mean yield of the bill hybrids shows a normal distribution and is more than doubled that of the bill, the homozygote bill. Same is true for introgression lines and introgression line hybrids. So the bottom line is that if you're interested in yield-associated trait, it's best to look at the bill hybrids, because the bills, because they are homozygote for genomic regions coming from the wild species, are not doing too well. Quite a lot of them are almost sterile. Next slide shows the LA716 bill hybrid. It's a analysis of total yield under drought conditions. And you can see all the QTL on the 12 chromosomes and showing the load value of the effect. And if you look at the load value of the effect, you could see that only on chromosome 4, the bottom of chromosome 4, there is a peak that is beyond load score of a threshold of 3. And if you look at this peak in the bill hybrids, there are 6 plants. If you look at the bottom part of the text there were 6 plants that were heterozygous for the pennellii segment in this region. The mean yield of them was 2.9 kilograms, whereas there were ones that carried the cultivated tomato allele the M82 allele had a higher yield. There were 351 of those, and a yield of 4.7 kilogram per plant. Even in a heterozygous condition, the effect is negative in that region. Next slide, please. Now, here you see the same experiment that was conducted for the LOST BILs, for the LOST BIL hybrids that Shai presented earlier. Here you can see on the top, you can see the effect of each genomic region from the 12 chromosomes on the total yield of the plant. And below you can see the load score, the threshold for that. You can see that in chromosome 9 and chromosome 10, there are QTL that affect yield, and these increase yield by 15%. You can see the chromosome 9 and 10 on the bottom, and the effect as seen on the top. And on chromosome 4, there is a QTL that affect yield, and it's also increasing yield. It's not very significant, but it's log 2, and it's increasing yield relative to the control, or to the mean of the population, increasing yield by 10 to 15%. So the bottom line that I want to stress here is that there is these two accessions that reside closely in South America, have different QTL for yield under drought condition. This we repeated 2 years, and the results are consistent in the 2 years trial. You can see that the chromosome 9 and 10 that have a significant effect on yield, and they improve yield, were not detected at all in the LA716. And chromosome 4 was detected in the LA716, but had the opposite effect. It reduced yield, and here in the LOST BIL hybrids, it increases yield. So it's I follow the same rationale or the same conclusion that Björn presented, that there is a lot of diversity in the species, and even in closely grown accessions. So it's not enough to say, okay, I screened Solanum pennellii, where you take a single accession and screen it. You see that in Solanum pennellii, there is tremendous amount of variation. The same is true for other the Solanum species. And that's why the Tomato Genetics Resource Center is so tremendously important for scientists working on tomato, and it should be there and provide the seed of the wild accessions, not only today, but for the next thousands of years for tomato geneticists. Next slide. Okay, so this is the final slide. Well, we have a problem now because we cannot, it's difficult to distribute seed because countries, people check the mail, and if you don't have full phytosanitary paperwork, the seed is destroyed. So what we are doing now in the framework of Harnesstom and also in the framework of another EU project called Capitalize, we are growing the tomatoes. You can see them. A picture. It's a picture I took 2 days ago. We are planting it. We planted it in a greenhouse of a farmer that works with the seed companies and is hopefully he will be sterile, and what he will do is produce self-seed of all the bills, of all the layer bills, and he will also produce hybrid seed of each bill cross to the each bill crossed back to the layer to get the 1,500 bill hybrids. This is a major project now in the lab, and I hope at the end of it, by the end of the beginning of next year, we will be able to send scientists the entire collection and the entire set of bills and the entire set of bill hybrids, so they could work on it independently. Well, thank you very much for for your attention, and I think it's break time now, huh?
Yeah, but before we would like to maybe to see what is in the chat, questions for you and the other speakers, if there is any.
I have a question for Björn. Can I didn't write it.
Okay, go ahead, because not everybody has a mic to speak that you have.
So Björn, we've been looking when we did the mapping. Are you there, Björn?
Yes, I'm here.
Yeah, great. So when we did the mapping, we used the SPET technology, and the SPET was designed to cover important genes in the tomato genome. So the SPET was designed based on the cultivated tomato only. Now, when we use the SPET for the mapping, we see that there are quite a number of large deletions of a few megabases or a few million base pairs that are present in the cultivated tomato, but not present in the Solanum pennellii genome. There are about, if we count all of them, there are about 1,000 such regions that we detected using 5,000 SPET markers. And I guess that if we do the opposite, if we use SPET design based on the pennellii, we would probably see much larger amount of pennellii specific regions. And I wonder, first of all, from what are the estimated genome size nowadays for the cultivated tomato and for the Solanum pennellii? And did you come across such or did you analyze your data to come across such present absent condition in the cultivated tomato or the pennellii?
Yeah, I would have to look at the exact ones. It's still that pennellii is larger, but that's not necessarily genes, because that is mostly transposons. But yes, there are regions which are missing from one or the other. We are looking as I showed a little bit gene by gene basis. And actually, together with Charlie Underwood, we also have an updated LA716 genome soon, where we did some of these analyses together with them. But it would be good to share some results that would be quite interesting. But yes, there's genes also in Solanum pennellii which are not in the cultivated tomato.
So we
And
can
there-
send you all the positions where we find sequences, but that's only relative to the lost accession. But I'll send you all the sequences, all the regions where we detected the absence of sequences from the pennellii genome. Maybe it will be of value for you in the future.
Absolutely, yeah. We also have, because we have resequenced LOST again, and also I think a little bit of LA2963 with long reads now, because since we simply just grabbed everything from the RIC center and are sequencing that, or did sequence that, we also have a bit updated genome versions for these.
Thanks.
Thanks a lot.
Any other question for Dani, or we go for break? And after break, we will have a Shay Trigerman talk. Dani, I have a question. So there are traits like yield and stress or drought condition, maybe under the control of many different genes or loci. What is your impression? How many are necessary to produce tomatoes in a good yield under such a stress conditions?
Well, you know, tomato, I learned from Alan Stevens, who was a great tomato geneticist. And he said tomato is like a sponge. The more water you give it, the larger and the heavier it becomes. So there's no way to get a very large tomato with little water. So it's not realistic. But I think of deficit irrigation, you know, in places where instead of giving, like in Spain, where you have drought conditions or water is lacking, then to select for varieties that receive, let's say, 75% of the amount of water, and see if under such conditions one can find varieties that produce similar yield as in irrigated conditions. So if you save even 15% of the amount of water, it's valuable. Also note that if you irrigate less in processing tomato, then the bricks, the sugar content, increases. And in processing tomato, the yield is not just the fresh yield, it's the yield is how much tomato sugar are harvested per unit area. So if you don't irrigate as much, and you use varieties that are adapted to drought conditions, then you might obtain the same yield. The problem is that people are reluctant to try it because farmers, you know, they pump the water, they push the water in, and they because they get the bricks doesn't play much role in payment. Whereas fresh yield is a major factor. So it has to be some project that like a national project where farmers would not suffer from the experiments of scientists, and in the long run they would benefit.
Okay, clear. Thank you, Dani. If there is no any other question and there is none in the chat, we will take a break to coffee or pee and get back into the room.
Maybe, Toni,
sir.
maybe you can make sure with Shai that
Yes. She confirmed over the
first
chat
time,
that the presentation is ready now for her.
Okay, okay. Okay, have a nice break.
See you. Simo, please, or Dave, please put the slide that we organized so that people can have this rather than a black screen. Um, Hi, Simo. Please stop sharing your screen and we welcome you all back to this session, and we will continue with Shay Trigerman. Shay, are you ready for your presentation?
Yes, let's make sure that
Okay.
it is indeed the case.
Okay. So Shay, I remind you, is going to talk about the genetic mapping of the forked inflorescence. Now it looks different, so it's good. Let's see, move to the
Wait,
slide.
wait.
Now, presentation mode.
Okay. You can see?
Yes,
Yeah.
it's
Yes,
working. It's working.
finally. Great.
Thank you, Shay.
So happy. Okay.
Please go ahead.
Okay.
So I'll start where I left off before. So just so you see what I'm talking about here with the fork inflorescence. In this part of the screen, you can see a regular inflorescence with one branch. And in the center is the fork inflorescence, where the branch is splitting into two in this case. And finally, the compound structure that is highly branched and bears hundreds of flowers and fruits. Okay. So for the mapping of the gene, we chose the LOST BILs population for several reasons. So first, it has already been sequenced by SPED technology using around 8,000 markers across the genome. It is also viable for biparental QTL analysis. And there has been previous work in our lab done by Shai Torgeman, who already identified an association of chromosome 3 specifically to the fork structure in the LOST BILs through phenotyping and QTL analysis. And that is great for me because it allowed me to focus only on lines that contain the pennellii introgressions across chromosome 3. So instead of looking at all the 500 lines, I had to work with only 426 lines that are relevant to my study because I know already that they have introgressions in chromosome 3. And these lines were planted in the fall of 2022. So that preliminary information that focused us on chromosome 3 led us to look at the other genes related to inflorescence that are found and located on that chromosome. So some of them are falciflora that controls flowering time, SFT, single flower truss, that regulates the transition and maintenance of flowering in tomato, and also fruitful 2 that regulates transition to flowering and also the branching of the inflorescence. So for the phenotyping of fork, I created a qualitative variable to describe the spectrum of the trait. Now, since the phenotyping is done on plants and not on each inflorescence separately, I decided to set my scale. So I started from 0, which means no forks at all, to 4 or 6, which means that about half the inflorescences are forks. Finally to 8, where the plants exhibited inflorescences resembling compound. So it is important to note that fork is a dynamic trait, meaning that the structure can change within a plant from inflorescence to inflorescence. So it could be a plant that has fully regular inflorescences or fully forks, or some regular and some forks, etc. And in this picture, actually, you can see the inflorescences of a single plant from the first one to the 6th one. And you can see they change throughout the plant's growth. So the plant started with regular structures and the later structures are forks. And this, in this figure by Zhang, we also see the dynamic nature of the trait. And here they present the effect of inflorescence position and different growing seasons on the variation of branch number. of 10 inflorescences throughout the plant. In this case, the first inflorescences are highly branched, and the number of branches decreases in the later positions. And just as a quick side note, inflorescence in general is a very dynamic trait, not just in relation to branching. So, for example, here we can see that the arrangement of the flowers on the branch can also change throughout the plant. So this is a regular inflorescence with fruit on either side of the branch. And underneath it, on the same plant, is an abnormal inflorescence where the arrangement of the flowers, or the fruit in this case, is mostly on one side of the branch. And we termed it half moon because of its shape. Okay, back to fork now. After phenotyping all 426 labels according to the qualitative scale, I performed a biparental QTL analysis and found an association between the trait and a specific marker on chromosome 3, of course, sitting at around 64 million base pairs. And from that, we derived that the gene for fork is somewhere assumingly near that site. So I looked at the chromosome 3 map of the bills and the area near the marker, and I also opened my browse, and we noticed that the fruitful 2 gene that I mentioned earlier is sitting very close to the marker, in fact, about 700,000 base pairs away. And as I said, fruitful 2 is known to regulate the branching of the inflorescence. So that assured us that this is the suspected area we want to focus on. I looked at zooming in on that suspected area allowed me to examine more interesting lines. So for the next planting season, I selected lines that either have an introgression of loss that fully covered the suspected area, or lines that were recombinant in that area, or lines that did not have the introgressions at all in that area. So a total of 42 lines. And they were planted this summer, summer of 2023, and phenotyped again. And I designed the PCR marker for fruitful 2, which was the suspect in, one suspect, and performed the t-test. And I found a significant association for fork, an even better one than that of the spec marker. And if we look at this little table here, you can see 20 plants carrying the lost allele in the fruitful 2 gene had fork inflorescence, as we expect, and 15 lines that carrying the allele had regular inflorescence, again, just as expected. So fruitful 2 definitely looks like the prime suspect, but there are some exceptions. Like you can see, 3 plants carrying the lost allele had regular inflorescence, and 4 plants carrying the Lea allele had fork inflorescence. So there are some exceptions. So the next step would be a fine mapping of the fruitful 2 gene. But at the same time, looking at the genome in the area near the fruitful 2, I noticed a MEDS-box gene very close to the fruitful, in fact, just 1 gene away. And it's called MEDS1 or EJ2, enhancer of jointless 2. And this is a MEDS-box transcription factor that allows, that belongs to the Sapetala subfamily. And the transcripts mostly accumulate in flower and fruits. So it could potentially also be related to our phenotype, the fork phenotype. And definitely it's worth investigating further, which is what I'm going to do this coming season, in hope of identifying the best gene to associate to the trait. Maybe it's not fruitful, maybe it is, who knows? Now, it appears that fork in general has the potential to boost up yield, right? So more branching, more fruit, larger yield. And I've examined it this summer in a certain genomic background unrelated to the bills. but I couldn't reach a clear conclusion. There seemed to be, like you can see here, a sort of a linkage drag to a gene that lowers productivity. So you see the more forks there are, the lower the yield, in this case, in this family. But it was inconclusive and not consistent with different families I've analyzed. So this season, I'm going to be examining the effect again and maybe understand it better. So thank you so much for listening. Sorry for the issues before. Thank you.
Thank you. Thank you, Shai. And if you have any question, any comment to Shai, please write on the chat. And we're going to move to the next presentation. Will be by Saleh Alseekh from Max Planck about BIL fruit metabolisms. Saleh, can you share your screen?
Okay, yeah. Can you see the screen?
Yeah, we see
you hear me?
We hear you clear and we see the screen. Perfect. Yeah. The presentation.
Okay.
Yeah. Good. Okay, go ahead.
Okay, so yeah, thank you very much. First of all, hello everyone, and thanks again for the invitation just to present some of the work we're really doing on the bills, specifically the LOST bills. I will just show an overview and tells one story we're really doing in the lab. But first of all, why we really care about metabolites or metabolomics, just really an overview. So we know, or it has been really reported that plants, they really produce large diversity and structurally metabolites that some of them contributed to the growth and developments. And the others also, some of those ones related to the fruit quality, like the volatiles, lycopene and vitamins. While the majority of the compounds, they are really called secondary metabolites or specialized secondary metabolites. They are really involved in the interactions between the plants and the environment that we know. Plants, they are really settled and they are not moving, so they have to have really a strategy to protect themselves from the surrounding environment. But also metabolomics, they are really, involved a lot of metabolic engineering and also some of those compounds really. Sorry?
Se che è peccato il prima, così siamo ricoverati.
Please go ahead.
Okay, so yeah, so again, some of those compounds as well really known to be involved in the healthy benefits like antioxidant activity or anti-cancer. So in the lab, we are really doing the forward genetics and basically looking at the natural diversities between the wild species or within the accessions of the tomatoes in tomatoes or also other crop species and integrating genomics, metabolomics to do a gene discovery at the end. As I mentioned, metabolites is really quite diverse and complex, and we are really interested mainly first in some of the compounds involved in the fruit quality, like sugars, amino acid, and organic acids. But also other important two kind of compound classes is what they call the flavonoids and the steroidal glycoalkaloids. And today I will just show one examples involved in the sugar metabolism and fruit quality at the beginning. So I also want to mention that, so we have been really working with the biparental populations or the crosses between the wild species and the cultivated and collaborating with long time with Dani Zamir using his population. He already mentioned the 76 lines, but also the 400 bills from the 716s. The Noriki also, this is also one of the population produced in that lab. Also, we're still really using these ones. And also the GWAS from there. And recently, of course, the LIA and top bills, the one related to this workshop. And yeah. So for what we have done so far for the bills, I mean, this is most of the work done by a PhD student in the lab. So Isra, she's really did most of the work. And we got really fruit materials, but also leaves materials from several experiments done in Dani's field or greenhouses. And I'm not really going to go through all of these numbers, but I can tell you about 5,000 samples already processed and analyzed in the lab so far. And mainly we are really looking at general profiling. It's targeted and non-targeted profiling. The LCMS, the secondary metabolites or the specialized metabolites, and the derivatized compound by GCMS will quantify the sugars, amino acid, and organic acids. But also we have really platform for the lipids where we measure the lipids and also some of the carotenoids, and with some work as well. So we follow up down to select some candidate genes and do some molecular biology for validating some of those ones. And again, today I will just give an overview and one story about the sugar metabolisms. So for the overview, this is what I can really say about the number of the compounds we have really identified, or basically confidence about the identifications of the compounds when we looked at the LIA and the top population. So we have about 80 primary metabolites, or call it derivatized compounds, and about 700 secondary metabolites and 200 lipids with some pigments. If you look at the diversity between the LOST, we call it LOST from here, and also the LIA and the top, the backgrounds, these are some of the, the differences, and these are the F2s where also, it's quite also difference when you do this. And this is really the metabolite profilings in general, using all of these compounds in the LIA population. This is about 1,500 in one of the experiments, and this is really the top. And again, as everyone really does with using this, so you do the QTL mapping. I'm not really going into details for all of the QTLs and number of QTLs we have really identified and working on, but this is just a general overview. There are really thousands of QTLs identified by these two populations. This is, for example, the numbers here. I call it here a significant SNPs, but of course, we have to look deeply to identify really the real QTL, and this is from the secondary metabolites. These are the lipids, and just here showing, number of SNPs or significant SNPs for the compounds we really measured. And this is really the GCMS data, which is the one I will just show or follow more in this talk. So these are the LIA QTL mapping on GCMS data, mainly primary metabolite, the sugars, amino acids, and organic acid, and these are really the top. So there are really some overlapping, of course, and clear overlapping between the population, but of course, there are really other unique QTL. You only can really see them in one of the populations. So again, one of the things I'm pretty sure this is really Dani already done with really other phenotypic traits, but also when it comes to the metabolites, the first thing you would like really to look at is really a proof of concepts and how this really population different from the previous populations we have really profiled. And here I'm showing one trait or metabolites, phenylalanine. This QTL have been really identified from other populations before, and it's really clear from the 500 top and the 1,500 tops. This QTL, the major QTL, is here. And if you look to the breakpoints and the recombination, I can guarantee that there are really less than 5 genes, or in this region. And the best SNP really mapped to the PAL gene, which is really the first, is the main enzyme at the first step of the penin-prepanoids pathway. As I mentioned, we know this gene before. This is really cloned with the Nuriki population, also really developed in Dani's lab, and combining with the 76 lines and 440 BILs line from the 716. So we know this gene and we confirm it by VIGS and molecular analysis. So other traits as well is really known. For example, also we measured carotenoids, and this is really known gene as well a long time ago from the population. So it's also clear QTL mapping to the previously cloned and QTL genes from this. So, but the question now, what is really new, what we can really do with this new population? Are there really any novel QTLs? Yeah, beside, of course, in addition to really the higher resolutions, but can we detect QTLs or from using this population that never or not really previously identified? And this is really one of the QTL, Isra in the lab, she's really following in her PhD. This is for the sucrose, and this is one of the main, it's a sugar metabolism. And this QTL, these are from the homozygous lines from the Leah populations, and these are really the top population. Both they are really mapped to the end of the chromosome 12. It's again, because also we have really the heterozygous lines from the top populations. Looking at this QTL here, I mean, there is really a QTL here, but not really passing really the threshold we have here. And I remember when I sent this to Dani, and the first question he asked is, what about the bricks? Because, talking about sugars and so on, And we know, of course, I mean, this is really, I copied from, or at least we used the data from Shai and Dani recently published. And we showed here, this is the QTLs for the bricks, and there's no QTL for the bricks on chromosome 12. So, but if you take really the bricks data and you correlate it with the sucrose, this is really the correlation between the bricks and the sucrose here. It's really about 90% correlated. It's very well correlated. So I think we have really a clue why this, or we don't really see bricks QTL here. However, this is still really to confirm, and we will follow up. So anyway, so to find the gene or genes, what is really kind of candidate genes that could be behind this QTL, if you look at this region, I think there's less than 15 genes or like this, looking at the recombinations here. And in this, there is a cluster of 3 genes coding for invertase inhibitors. So again, and also we really looked at really the other data and so on. I mean, if you go back to the expression data, this is a fruit expression data from the 76 line from Jim Giovanni. And the expression of one of these genes here, I'm just showing the expression data is really, there is really quite a strong expression on 12.4, which is also really located within this region. And if you look at the expression data from the leaves of the 76 integration lines, there is no expression or even less in this line. And if you check really the EFP browser, comparing M82 and the pennellii, I mean, this gene is really quite highly expressed in the fruits and in the wild species, the pennellii 716 and not. But again, I mean, we are really looking at the LOST here, and is really, this is something just because the expression and why we don't really see this QTL in the previous population, because we already have the data and profiled the BILs population from the 716 and the ILs, and we don't really see any QTL for the 1214 within this region. So, but in general, just a general information about the tomato fruits. So we know the wild species, they are really accumulating sucrose. And this is really the GWAS panel. I also mentioned at the beginning, we have really data on about 600 accessions here, including 60 wild species. And if you look here, these are the wild species, they are really having high amount of the sucrose comparing to the other cultivated lines here. And if you look at this here, you can really see the sucrose comparing to the seriformes or the cultivated is quite higher comparing to the, glucose and fructose. Tomatoes, they are really usually or known, it accumulates, or the one we eat is the fructose and glucose and citric acid. These are really the major compounds there, or besides some other organic acid like glutamate and malate. So again, also this is really something, it's just by chance we went to, it's really clear this is from the two populations and more than one experiments. But again, we have also one harvest or one experiment also done, or we collected samples, or Dani sent samples on about 50 selected bills. These are really with the more replications, with the 3-4 replications. And when we did the metabolite profiling, so this is in both leaves and fruits, we can really confirm that this QTL is really a prominent and strong QTLs back again for the fruit, but not in the leaves materials. So this is really clear. In this case as well, we saw some signals as well for glucose and fructose because, expected sucrose will convert it by invertases to the glucose and fructose and then contributed to the bricks, for example, or total soluble solid. So when Isra also started working with this, so what we did as well, so we amplified the targeted gene within this region, or from the LOST, from the LIA, and from the pennellii LA716, and these are really the polymorphism or the sequence, the one we really amplified and sequenced here. And of course, there is really a clear difference. For example, the one in the middle here, this is really the pennellii. This is quite really different, but they are really different. Also, polymorphism between these are the reference. I think the Heinz, maybe Isra also she can comment on this. She's also within the audience, and there's also other differences between the LIA and the reference genome for this really gene. So we are really investigating this a little bit more. For the localization or the expression or the proteins, we can really see in the cell wall. However, Isra also doing now hairy root transformations really to confirm is this is really the one really in the cell wall or the vacuoles, because there are really other studies as well showing the expression in the vacuole, not in the cell wall. So we Isra also generated stable overexpression lines by overexpressing the 3 alleles in the moneymaker in the cultivated tomato here. And this is really some preliminary result. I will go very quickly now on T0 plants. They are really, of course, kind of really changes. These are the metabolite data on the fruit materials. There are really some separation between the alleles here, but what we can really see here, the loss, they are really overexpressing this gene is really highly accumulating the sucrose. The pennellii, they behave a little bit different, so we still need to confirm by the QRT-PCR the expressions are these are also is a negative or positive, and see to confirm this result again. But we can really see here overexpressing the loss is really clearly higher accumulation of the sucrose in the fruits, and no changes. We don't really see really a very clear changes in the leaf of these materials. There's one experiment now. It's really growing in the lab. We can really growing in the greenhouse, and we can really see also this really influence the yield. So the yield is really significantly low by overexpressing the lost allele in the cultivated really tomato, and where there are really no other significant changes, for example, a number of fruits or flowers and so on. And metabolite profiling, I mean, this is the T1, they are really in profiling really to confirm these results. And in this case as well, we really collected as well materials from different developmental stages and leaves to get a comprehensive overview about the validation of this gene. And finally, so also, I mean, within the work as well, we are really following really the same approach. Also, Shai and Dani did in their recent paper, and we did also epistasis. This will be also, included in this work. And these are some of the examples. You can really sucrose here with a really more interacting or epistatic interactions using this population. This is really still in progress, and we do it as well with other crops. This is really just one story, and of course, we are really selecting really many other QTLs and interested metabolites to follow up. For example, a secondary metabolite with several genes. Mainly here, we are really focusing on transcriptional regulations and using VIGs or hair root transformations to validate those ones. And there are really others related to the lipids and also some hormones or jasmonic acids. This is really also SRA-generated CRISPR-Cas9s for these interesting QTLs. It's also confirmed for several experiments. It's really mapped to the logs here. A cluster of the logs here, and we know this one really involved in biotic interactions. There are also really some phenotype. This is a T0 from the CRISPR-Cas9 experiments, plants that are growing in the greenhouse now, and we will see later if this really will validate and how this really link to the jasmonic acids and the biotic stresses in this population. And finally, I mean, hope once we really finish this and publish it, this will also be included in a database we are really creating now for really, in general, other crop species, but tomato will be having really the large majority of the metabolic profiling here. In this database will be focusing on the metabolites. So we're using all the population have been really profiled in the last years or so on with several experiments, but also we are including other crops we are really doing in the labs like pepper and quinoa. And of course, the project with the legumes that recently we got on the GWAS panel. This is, I think, what I have. And thanks again for invitation and also listening. And I would like really to thank Isra really for doing all the work here and the collaboration, especially Dani and Shai for really providing all the materials. Yaniv as well, and Leah for also helping collecting and bringing the samples to Golem here. And I'm happy to have any suggestion or questions, and maybe if it's not now because the time, I'm really happy to discuss this or receive comments really later. Thanks.
Thank you very much, Ale, for your presentation. And we have some questions in the chat, but probably we will go to these questions after the last talk of this session.
Okay, sorry, I need to stop sharing.
So you may stop sharing. I will go to the next one.
Where I stop.
That will be Edan, Rochsar, and Aviv Dombrovsky from fine mapping of TM1, a gene from resistance to the tomato brown rugose fruit virus.
No, I'm still sharing. Yes.
You are still sharing. You need to
find the red tab. The red tab on top.
Okay.
Okay.
Finally. Okay.
So, Edan, are you going to present? Okay.
Yeah, I'm here.
Yeah, it's okay. Yeah, we can see your presentation. So
yeah, we can see. Okay.
And we hear you perfectly. So yeah, go ahead.
Okay, thank you. So my name is Edan. I'm a PhD student in Dani Zamir's lab with collaboration with Aviv Dombrovsky. And as you said, I will talk about mapping resistant genes for tomato brown rugose fruit virus, or in short, TBRFV. So tomato plant is a very important crop, a very popular model plant, and is popular in consuming of fresh fruit. A little bit about the production of the tomatoes. So we can see here the production from the 1970 to 2021, where the red line represents the tomato production in tons, and the blue line represents the area harvested in hectares. A couple of interesting stuff we can see here. First of all, there's an ever-increasing demand for fresh fruit throughout the last 50 years. Another interesting stuff is we can see that up until the 2000s, the yield was very consistent with the area that was used. Around the year 2000, they started the use of F1 hybrids, harness the power of sorry, just a second. Of heterosis, and we can see better use. We can see the line of production, which is across the line of the area across. We can see higher yields from individual plants. To put it a little bit in numbers, so we have the worldwide production of tomatoes in 2021 was estimated at 198 million tons, which was grown over 5 million hectares worldwide. The leading producer countries in descending order were China, India, Turkey, USA, Italy, Egypt, Spain, Mexico, Brazil, and newcomer Nigeria. Just a second. So if we talk about the problem that we have, we're talking about the tomato brown rugose fruit virus, or in short, the TOBRFV. It was first reported in Jordan and Israel in 2014. It's a member of the tobamovirus genus. This genus is characterized by a long rod shape. It's a single-strand RNA plus sense virus with a genome length of around 6.2 to 6.4 kilobase, encodes to 4 proteins, highly transmittable. Its main ways of infection include the mechanical way. We can see here a greenhouse in protected crops. And part of the work in the greenhouse, you need to cut the leaves, and you can see if you have one infected plant, you can transmit it from to the entire row by using of the scissors or by workers' clothes. Another way of infection is by pollinator. It was shown that bumblebee, that is a highly used insect for pollination in commercial greenhouses, can also infect plants. If it lands on an infected flower, goes to a healthy plant, it can infect it as well. And if we're talking about seed companies, mostly they have a big problem with the preservation of the virus in the seed coat surface. Also contribute a lot to the transmission of the disease cross countries, cross continents, pose a very big problem for growers around the world. The member of the tobamovirus genus includes the TMV and TOMV, but unlike those two, or the entire genus, the TBRFV can break the resistance of the TM22 gene that is almost the only gene that is used for this type of viruses. Let's talk a little bit about the disease. So the symptoms typically develop within 12 to 18 days post-infection. We differentiate between leaf symptoms and fruit symptoms. The leaf symptoms usually composed of yellowing and the narrowing of the leaf to the point of stringiness, if you can see it. And if we're talking about the fruit, we have discoloration, and in very hard cases, we can even see the brown rugose fruit, hence the name of the virus. It's such a problem that the yield loss can vary between 10 to 70%, depending on the climate condition and the tomato varieties. We can talk about the current distribution. It's a lot of countries and many more that are still haven't officially declared. Countries marked in red are one of the 10 big production of tomatoes, hence the problem in arriving at the destination of the yield required for food security around the world, and a very economic problem for growers and and supermarkets around the world. If we're talking about ways of coping with it, so there is no chemical solution for it. So solution today based on phytosanitation. If we're talking about throughout the growing season, so we can see the protective gear, boots, all this huge condom that people wear a lot these days. A lot of using Clorox, sanitize the shoes and working tools. In between growing seasons, we can see a lot of soil disinfection and area and surfaces and areas in the greenhouses. And also, if you're in the seed business, seed disinfection, which is seems to be very hard, especially with the new requirements to pass a PCR test, which can also detect virus particles, which are not viable, but still it's a very hard issue for many companies these days. A different approach to handle diseases in general is the use of wild species and incorporate of resistant genes into cultivated lines. So I brought here some examples of tomato diseases, the resistant genes against them, and the corresponding wild species that they were originated from. If we're talking about the tomato virus, we can see 3 genes that were in use. The TM1 and TM2 originate from the Albocaitis, and the TM22 from the Peruvianum. The TM1 and TM2 were resistances were break by the TOMV in the 70s. So the main gene in use today is the TM22, which was broken by the TBRFV in 2014, and ever since no solution was found. So the research aim and what I will be talking about is we would like to screen for TBRF resistance in wild and commercial germplasm, and if we'll find a suitable source, we would like to identify the QTL or genes that are responsible for the resistance. In order to do that, we would like to, first of all, score the symptoms of the disease. So we created a scale from 1 to 4, where 4 was the symptomless and 1 was the most severe symptoms. We differentiated between leaves and fruits, and we used that score. And for our experimental system, what we wanted to do, we wanted to select germplasm to evaluate, sow it, and then inoculate it, either in the greenhouse, in pots, or in the trays. 4 weeks after inoculation, we wanted to evaluate the symptoms and to test it in ELISA, as we can see, when the sample is colored, we say it's positive and the plant is sick, and when it stays transparent, we say the ELISA was negative and the plant stays healthy. So we will be looking for the healthy ones. So to address the first aim, to screen for resistance in wild and commercial, we create a panel of 45 accessions from the wild and evaluated those. You can see the accession number, the species on top, and the score. So just to remind that 4 was symptomless and 1 was very severe symptoms. And already just by looking at it, we can see a few sources that are symptomless. But once we did the ELISA test, we only found 3 that were also negative in the ELISA, including 1 Chilensis and 2 pennellii. Just to put it in a table, maybe it would be easier. So the Chilensis accession was the LA1938, and the 2 pennellii were the LA5240 and the LA716. We wanted to do the same by screening in cultivated germplasm. We tested it in the greenhouses. We had in blue the symptoms of the leaves, and in orange the symptoms of the fruits. And the graph is arranged by the symptoms of the leaf. So we screen around 56 cultivated tomatoes, and I will focus on this area. We can see we found two cultivars from the UC Davis collection, one from the G2P collection, and one from Dani's private collection, which we named Rotem, that were negative in the ELISAs and showed no symptoms. So once we found a few suitable sources to work with, we wanted to identify the QTLs and genes that are responsible for it. So to do that, luckily for us, we had the backwards inbred line fields mapping population that Shai presented to you. It was originated from the pennellii LA5240 that was symptomless and negative in the ELISA test. We used the Lea population that is composed of 1,500 lines, where each line contained an average of 87% cultivated genome and 12% wild genome. The population genotype was determined using the single primer enriched technology, the SPET, which has a cover of around 8,000 genetic markers throughout the entire genome of each individual of the population. So to do so, we selected a panel that Shai presented before. He had 60. This is the first time we used it. We only used 53 lines that cover the entire pennellii genome. You can see here the SPET marker on the X from 0 to 8,000 with the corresponding chromosome on top, and the individuals and their genotype, where green is the S. pennellii and the red is the cultivated genome. So we wanted to cover the entire genome. So we planted those and inoculated them and did a scale of the symptoms. And what we found is that based on the disease symptoms, we mapped the resistance to chromosome 2. We have one high peak of around 4.5 on chromosome 2 on an area of about 13 million base pairs. It's a start, but it's not enough. So what we wanted to do next is to zoom in on chromosome 2. This time we used the 72 lines as a panel that covered the entire 13 million base pairs. Inoculated those lines, and ELISA tested all of them. And this time we were able to reduce the interval to about 1 million base pairs between those locations. We thought we can do better, so we wanted to do fine mapping using recombinants. So this is the 1 million base pair interval. We added some markers in between them to higher the resolution. You can see in both ends are the parental lines, the cultivated line, and the pennellii. We have the ELISA results. And in between them, the 7 recombinants that we use for this test. And what we can see is that the marker in this marker on the 34 million had the highest correlation with the healthy plants. Everyone that had it was healthy. Maybe it is easier to see that this line and this line, that the recombination happened somewhere before and after this marker, were both positive, were both sick. And the 3 lines that had the entire introgression, or this marker in particular, were healthy. So what we can conclude from that, because we don't know whether the recombination event occurred exactly, that the resistance is somewhere between the 2 neighboring markers next to this marker. So it's an interval of around 500,000 base pairs. Next thing we wanted to do is to search for candidate genes. We used NCBI genome data viewers and search for the genes that are known in this area. We found that there are 32 genes. And if we look at the list of the genes, we can see two that might be interesting to us. One of them is called hypersensitivity related 4, and the other one is TM1, which is known to be resistant genes against the virus family in particular. So that for us meant that it's the leading candidate for us. So just to summarize, we identified several suitable candidate sources for resistance from the wild and cultivated tomato. From the wild sources, we found 3, the Pennellii 716 and 5240, and one Chilensis, the 1938. From the cultivated lines, we found Rotem, 2 from Davis, and a G2P cultivated line that were resistant. We chose to continue with the mapping population that we have for the Pennellii. And the mapping of the resistance was conducted using the BIL population from the it was symptomless. The resistance factor was mapped to the genetic location between the 33 million and 34, around 500,000 base pair interval on chromosome 2. And searching the literature, we uncovered 2 potential candidates. One is the TM1 and one is the hypersensitivity related 4. Again, we are more trusting on the TM1 on that case. And that's it, just to say some thanks to the Zamir's lab, Dani, of course, Kfir and Shai, for the Dombrovsky lab, to Aviv, Chen and Yelena, and to Amnon from Krishtil. Thank you.
Thank you very much, Edan. Very interesting presentation for this problem that we have now in the tomato community, for sure. Yeah.
Thank you.
We have some time for questions. I have. Yeah, let me open the chat. Where is the chat? Okay. We have some question for Shai. One question is, what is the level of branching you observe in your BIL family? In other words, what is the distribution of fork clusters versus highly branched clusters and versus normal clusters? You may find, Shai, the question in the chat.
Yeah, so highly branched lines are rare in this population. I spotted about 15 lines out of 400, and most lines are regular. And there were about 130 that were somewhat forks.
Okay, next question also for Shai. What about the total number of inflorescence?
Okay, so in the fall of 2022, like I said, I used the qualitative variable method. However, I did feel that it should be more accurate. So this summer, I tried scoring the plants with percentage of forks to get a more precise score that neutralizes the effect of the number of inflorescence. So for each plant, I looked at 10 inflorescences randomly and calculated how many of them are fork. So I hope I understood your question correctly.
Yeah. Okay, we have another question for you, Shai. How did you deal with the dynamic inflorescence structure in the same plant? In which way you score it from the same plant or same accession? And how did you analyze the data?
Okay, so first I looked at specific plants, and each plant represented a specific line. And again, I used the original variable was qualitative. So if I'm looking at a whole plant that represents the line, if some inflorescences are fork and some are regular, I gave it the score between 2 and 6, depending on the number of forks. So 2 was less forks, 6 was more. And of course, the score 0 is fully regular. And again, the current variable I'm using is based on percentage, so I think it sort of relates to the dynamics and neutralizes it. And the statistical analysis, I use the RStudio with specific QTL packages that allows me to cross the spec markers data with the phenotypic data that I collected to find associations. And it's similar to GWAS, if you're familiar, genome-wide association studies, except it's under biparental terms. And also I use the t-tests, standard t-tests for the analysis of the PCR markers.
Okay, thank you. We have a question for Saleh. What is the correlation between the sucrose and the fruit weight?
Yeah, I think it's really a good question. So we haven't really done this yet. We will do, and we will see. But yeah, that's, there are really always the negative correlation between bricks and fruit weight or size. But this is a good point. We will check and see if there's something within this.
Okay. I have one question for you too.
Yeah.
The QTL that you find for the metabolites, usually you find it's a structural gene that is involved in the pathway behind the QTL, or you find or you don't find any, let's say, structural gene involved in the pathways, or you have some kind of percentage, or
yeah,
or what often is this?
Yeah, so yeah, in general, I mean, if you talk about the metabolism in general, yes, we can really see regulations like the transcription factors in general, especially when you talk about secondary metabolites. There are really a lot of transcription factors, and that's one point. Actually, we're following up with the secondary metabolite part. We are really focusing on more transcriptional regulations, but also we can really see a lot of structural genes, like the one cloned before, but there are really other known ones as well. We are really also doing part on sterolide glycoalkaloids, and we compare with other ones. So we identified as well some structural genes for one not really identified so far. Yes, but we can see both. Yes.
Yeah. Thank you. We have two questions for Edan. The first one is, given TM1 linkage with longer internode length?
Not that we know of. We did not see any other characteristic of the plant affected by the TM1 presence. Depends on the background.
Okay. And the other question is, the QTL in chromosome 2 is enough for full resistance, or additional loci are needed?
So the TM1 is the main gene that is needed. We are now continuing to evaluate it and see if there are any QTLs needed. My guess is there are other QTLs that lower the resistance, for example, and not necessarily make it stronger. So you need to check on different backgrounds and see what they does and what to get rid of, maybe.
Okay. Thank you. Any other question? We are still on time. Thank you, all of you, to keep track, to keep the time, and for your beautiful presentations. And I think we can do, if there is no more questions, we can make a break and we come back and to 11:45. Okay. So see you in 3 minutes. 15 minutes. Yeah. Joder. Toma, toma, toma. Parla. Parla, parla.
Hello.
Hi, hello, welcome back. Before you start, Amara,
Yes.
I wanted to ask Simo, can you I would like to ask everybody to switch on the video cameras so we can take a picture of all together, okay?
Okay.
Like for the memory of this, I mean, for the record. So please, if you activate your cameras, then Simo or Dave,
Anyone who knows how to do it.
or anybody who knows how to do it.
Vista. Galería.
No,
Okay,
no, I learned. This is easy. Okay.
Okay. So, cheers.
Cheers.
Okay, I want to take another one for the
next
other
page.
people. Okay, thank you. Some that are still shy, that they don't open the camera. Wait.
Okay.
And that's it. So it's for the size. Shy.
So we are now 112 people connected, which is very good.
Okay.
And we are back.
Okay.
So, Mara, the floor is yours.
Before I start, there is a question, still a question from Gancho to Edan. I don't know if Edan wants response to it. He said that it's about the stability of TM1. He thinks that is not stable in Lycopersicum esculentum, in Solanum Lycopersicum.
Yeah, no, that's a very good question. So the last question they also asked me, and we do know that there are some genes that lower the resistance effect that may come from the cultivated tomatoes. Also, the way that the TM1 works, it's unlike the TM2, it doesn't cause HR, no cell death, only inhibition of the virus replication. So if there are changes in temperature, for example, they could affect it. If there are other diseases, can also affect it. We are now trying to understand which factors affect the resistance, maybe make it stronger or maybe weakens it.
Okay, thank you. Thank you. So let's move to the last session of this training class. And we will start with Yaniv Semel that will describe his Phenome networks database. Please.
Hello. Hi.
Hello.
Hi.
Please share your screen.
Yeah, just a moment.
Yes.
Can you see my screen?
Yes.
Okay, very good.
Okay.
Just a second. Okay, so first, thank you very much for the organizers for inviting me. So I'm Yaniv Semel from Phenome Networks, and for those who are not familiar with Phenome Networks, I will tell you that we are an Israeli company that develop software solutions for plant breeding. We work mainly with private companies. We work with about 150 breeding companies and seed companies. This is a partial list of them.
and work with about 3,000 users, which most of them are breeders and people who are involved in breeding. And our platform called Phenome 1, and it supports various management tasks related to plant breeding, including the germplasm development, pedigree, the experimental design of trials, and also the mobile application for collecting data. We have a module for data analysis, inventory-related tasks, and so on. Let me continue here. Within Harnesstom, we are responsible on collecting and integrating data coming from multiple sources, including data coming from previous projects, such as the G2P Sol, Traditom, Bresov, TomGem. All of them are projects on tomatoes. And also to manage and load data that is generated within the Harnesstom project in work packages 4, 6, and 7. In particular, our platform support what is called interoperability, which means that it's possible to integrate our system with external database and applications. This was done by implementing what is called breeding API. It's API, it's application programming interface. It's basically programmatic access to our system that is done using from external systems and allow or support creating integration to external systems. This is done mainly by our customers who are using these to integrate our system to their external system for many different purposes. But here in the Harnesstom projects, we could utilize that for creating this connection from the Harnesstom database to other external systems. So in this talk, I want to show you a few of the main features done or used by our users, breeders. The first thing is about phenotypic analysis, and this is going to be based on a new module that we have released recently and replaced as a previous one. The second topic is about Phenogene, which is an algorithm developed in collaboration with Gene, and this is, we call it kind of a process optimizer. I will show you how it can you can choose markers coming from different parental lines or different sources, and it will give you the most optimized way to create the crosses schedule in order to combine different markers into a single genotype. So let's start with the first one, the phenotypic analysis. I will go to my browser. I will show you a very quick demonstration of how it can be used. So initially, I will choose the crop. In this case, I will choose years that I want to analyze. I can choose programs, either one or most of them, and click on Next. It shows me the list of trials that found under this filtering, and then I go to the show results. And as you can see, I can just see the list of varieties. This module is mainly being used for product development or variety testing. So in this case, we have a short list of varieties or candidates to varieties that were tested in many different locations. I can change the trait. I can change the trials. I can remove trial, and as you can see, it's very responsive. So every trial that I choose to remove, it is automatically calculate the median and maximum and the quartiles, and I can choose all of them. I can remove some varieties, and they are automatically removed from the graph. I can decide to compare not the varieties themselves, but maybe compare between years, and then the comparison is among years. I could also compare between different countries. Here I have 2 different countries. I could decide to compare seasons, or even specific locations, or maybe just to compare all the trials. Okay, so and eventually go back to comparing varieties. And also the legend can change. So I can see color the data points by year. So you can see the year over here. Can remove year, but now the data points are color coded based on the year. It could be based on a country, and then this is Italy and Spain. It could be based on the season or site locations and so on. And also, in addition to that, I can use the remove outlier, the checkbox, to remove the outliers immediately. And this is very interactive and very responsive. I can change the criterion for outliers. This is based now on 2 standard deviation, but I can change it to 3 or 2.5 or 1 to make it more or less strict. So in addition to that, we have kind of a bar plot, which really perform kind of a statistical comparison. So you could choose a specific hybrid in this case, and it highlight in orange all the other hybrids that are significantly different. And again, you could change the significance over here, and then it is automatically being calculated on the fly. And just click on any other, or maybe make it less strict, and see the differences. So, and here again, you could remove trials, and it automatically calculated the mean for each hybrid. You could remove varieties, right, or add them all. So you could, of course, change the trait, right? And again, you could make the comparison with any of the traits. So that's a single trait, and maybe let me move to the statistical part. There are some tables which allow users to download the general statistics, the analysis of variant, the summary, and so on. So that's one aspect of it. I could go to the single trait and see the traits profile for all the hybrids. And again, there's a zoom in and zoom out over here. I can remove some trait or add them. I can make the calculation based on specific trials that I want, and automatically calculates based on the selected trials, and so on. So, and again, I can make the comparison not between varieties, but maybe between years. Okay, and see the traits profile across the years or across the countries. right? Or across specific seasons or programs or experiments, trials, right? So, so that's something that could be very nice to compare varieties. If I will move on here, this is an example of correlation. So I could choose the X and Y axis, and the correlation is automatically calculated. And again, I can choose different legends. So I could choose the year, and it's automatically color the data points based on the associated year or country or season or location and so on. Okay, so again, very highly interactive and responsive. And also there is a kind of a heat map that can use the same features. Another example is of a map where I can choose specific trials and see them on the map. So this is kind of a request coming from breeders to identify their location of trials. So this is the part of the phenotypic analysis. So that was a very quick demonstration. Let me move on to the second topic, and I will start with a few slides describing what we call Phenogene, which is a module, which is an algorithm developed in collaboration with Keygene that is try to collect favorable alleles coming from different sources into a single genotype. And this is an example where we have 4 possible parental lines. Okay, so what I'm trying to do now is explain the problem and how the algorithm works. So in this case, I have 4 possible parental lines, and on the left side, I have different disease-resistant markers. And as you can see, each line is resistant, like green, for some markers, but susceptible for the other markers. The same is true for each of these 4 parental lines. And my objective is to create a genotype that includes all the resistant markers within it, okay? And the algorithm will provide you the most efficient way to create the crosses until you reach the target genotype, which is resistant to all the markers. What we need, the data that we need, is the position of each marker on chromosome and the distance between markers in a centimorgan. Okay, this is the input data that the algorithm requires in order to calculate the number of plants and crosses needed for each generation until we reach the ideal genotype. And eventually the results will look like this. This is kind of a very simple example, right? With 4 parental lines that each one is resistant for a different marker, and the algorithm will tell you which plants to cross and how many plants are needed for each cross in order to get at least 1 plant with the required genotype in 95% probability. So this one, for example, we cross between these two hybrids in order, and we need 798 plants, okay, so that we can achieve at least a single plant. By the way, on average, you will get 2.9 plants, almost 3 plants that has this specific genotype. And eventually, you need to self it. For this, you need to have 81 plants, and eventually, you will reach your resistant genotype. So this is an example. Let me maybe briefly explain behind the scene how it works. If we have 2 parents with 2 markers, one of them is resistant to the first marker and susceptible to the second marker, and they are homozygous, we cross them to create the F1, which is then self and creates the recombinant gametes and parental gametes. So in this case, let's assume the 2 markers reside on the same chromosome. They have, like, let's say, in our example, 10 centimorgan distance between them. So the chances to get a recombinant is 10%, but we want the recombinant that is resistant in this case. So we need to divide 10% probability by 2 to get 5%. And of course, we need to have 2 recombinant gametes, so we raise it to the power of 2, and that would be the probability the probability to get a homozygous genotype. And this is, of course, based on only 2 markers. Now, if we have 10 or 20 markers, of course, the number of combinations is very, very high, and the algorithm does it for each pair of markers and try to find the most efficient way mathematically possible until and the minimum number of generations and plants until we get the desired genotype. This is a more complex example where we start with 5 parental lines, and it gives you the scheme. It shows you how many generations are needed. It shows you for each course of self how many plants are needed. It shows you the genotype in each step until we get to the desired genotype, and this is done in the most efficient way mathematically possible. Another way to utilize this algorithm is for hybrid reconstruction, where you define parents. So you can define homozygous parent 1 and parent 2 with your desired markers combinations, so that eventually you can cross them to get the hybrid. So this is the phenogene. Again, it combines favorable alleles into a single idiotype. It does it in the most optimized way, but it has limitations, of course. The first limitation is that it can be used up to 30 markers, because after then, more than that, it would create a huge number of plants and many generations, which makes it not reasonable for a typical breeding program. And of course, another thing that we need to consider is that it requires information about the marker's location and the distance between them. This is the input of the algorithm. And one of the disadvantages of it is that it provides you eventually a genotype and not the phenotype, right? So it can bring you the optimal genotype, but the effect on its phenotype, assuming there's epistasis or some negative effect between these markers. So this is something that we cannot predict, okay? Let me show you very quickly the interface behind that. So here I have an example where I have some parents data. So on the left side, these are the parents. On the columns represent different markers. I can assign markers positions to chromosome on this table. And in order to create a new project, let me create a test project in my example, and click OK. I get into this project. First, I will choose some parents. Let me choose all of them. I will choose also the markers that I want. Let me choose some subset of the markers. And the parents data, I need to define the ideotype or the ideal genotype. In my example, I will use homozygous one for all the markers, but of course, it's up to you depending on the marker. And eventually, you can run the results. Here, there are some parameters that you can configure. And eventually I can run the job, the algorithm. As you can see, I have an item, it's processing, now it's done, and I can click on it. And this is the results of the algorithm, where it tells me eventually here cross between two parents requires only one plant because they are homozygous. But then when I cross between this item and this, then I need 59 plants, and here I need more, and so on. So you can eventually see the cost, the total cost, or total number of plants and generations that are needed. This is the cumulative population that shows you the total number of plants needed and the total number of generations until you reach your resistant genotype. This shows you also the parameters that were used, information about the parents and the markers and all these arguments, and of course the results. So yeah, so I think if trying to summarize this algorithm, again, the main advantage is that it will combine the selected markers in the most optimized way. It will give you the best way to create this genotype. But again, we have to remember that you will get a genotype and not necessarily the phenotype. So it kind of assumes that these markers are very strong and can work in different backgrounds, or in different not different backgrounds, but that there's no negative epistasis effect between them. Yeah, so by this I want to conclude, and thank you very much for your attention.
Thank you, Yaniv. Very interesting presentation. You show very interesting tool, especially the one related to recombination is quite useful for all breeder, but also for other people working genetics as us. So you know, we ask people that have a question for you to write down in the chat, and at the end of the presentation you can get it. Thank you.
Thank you.
So let's move in the next presenter, that is Clara Pons, and she will talk about Harnesstom DB and tools.
Okay, let's share my screen. Do you see my screen?
Not yet.
No?
No.
I have a problem now. I suppose that I...
Yeah, try again.
Okay.
Maybe it's just slow.
Do you see?
No.
Okay, so I don't know what happens. I can share my screen. Let's see. Don't know why. Okay, let's try now. Okay, do you see my screen? Okay. Okay, so now do you see?
Yeah, yes, perfect.
Okay, so thanks to allow me to present the Harnesstom database and the tools. The database is still under development, so there are some things that need to still to fix, but there is a need for construction at one-stop shop database. In the last 20 years, there is a lot of data generated by different projects and different researchers that this data is not connected, stored in different databases or just in the computer of different labs. And to facilitate the use of this germplasm, we have developed, or we are developing, a curated open source multiomics database for facilitate the use of the potential of the germplasm collection. Our idea is to store all in the same place and make this data fair. The database is the Harnesstom is a relational database composed by 5 modules: the experimental module, the germplasm module, the phenotyping module, the genotyping module, and a variant catalog. The information that contains these modules are information about the crop, the programs, or the projects that contains the data, the trial, the studies, and in this all this experimental module, we will have find information about the experimental design, the growing conditions, the factors that are used to analyze or the parameters to grow the plants, the institute, the locations, and the contacts. In the germplasm module, we have information about germplasm. First of all, says that in Harnesstom database, germplasm is an accession in a project because the same accession can be used in different projects. We have also accession with the multi-crop passport description and attributes. Maybe we have some interesting features that we want to remark about an accession, taxonomy, synonyms. There are also collections. An accession can form part of a collection, crosses and breeding info, pedigree info, mapping populations, and image description. The database is also prepared to store some information about phenotypes as trait and observation variables. That is, observation variables are traits measured in different scales and methods. Also for store metabolites and analytes with information about the identification level and if it pertains to a health or flavor category, observations, experimental units, samples, and also, of course, the phenotyping values. In the genotyping module, have different capacity to store different genotyping platforms. It could be also could be a genotyping platform such as SolCap array, but also GBS raw data. And it contains information about the software, the marker that contains these platforms links to the raw data repositories, like the bioproject or biosamples in the ENA. We have also capacity to store variant set metadata and variant set and the genotyping values for each accession. In addition, we have included a variant catalog that is able to store metadata for QTL studies and GWAS studies, but also GWAS variants, QTL specific epistatic SFET or QTLs, and information about if the gene is cloned or not. All of this is standardized using a controlled vocabulary with different ISO standards. If they did not find any ontology, we create new ontologies for that. Actually, we have in the database information about the Traditon project, TomGem, and another project that is created mapping population to store different mapping population used in the projects that are created time ago. These have 6 trials and 24 studies. We have information about 2,000 gen plants that corresponds to around 2,000 accessions. We have recorded more than 10,000 accession images, 1 cross, 1 mapping population, and we have 170 gen plants with pedigree. We have metadata for 175 observation variables and 800 metabolites. But at this moment, the stored data for phenotypic values corresponds to 79 observation variables in more than 4,000 observation units that renders more than around 100,000 phenotypic values. In the genotyping module, we have 4 genotyping platforms, the Traditon GDS and the SolCap array in 3 genome versions, with more information about more than 100,000 markers. We have 5 variant set metadata, and you can download the VCF that with more than 2 million genotyping data for the accessions. In the variant catalog, we have 3 QTL studies and 8 GWAS studies, and we have around 800 variants, 21 for QTLs and 1,723 for GWAS. The controlled the ontologies are around 46 and ISO standard, and here you can see some examples that the ontologies that the database use. Okay, of course, in order to protect the intellectual property, there is some info that is restricted if you are not a user, or if the data has a, needs to be protected until published. In general, project trial metadata as well, GenPlasm, accession passport, and genotyping platform will be always public in Harnesstom, but all trial associated data that studies, phenotypic data, image, genotype, variant, could be public or private. We have a restriction. We can put the date for public release, and when the date is reached, data will be public. Until this moment, will be private. The database was conceived to have access through two ways. One is through a web application that we will see later, and also using an API access that is BRAPI compilant to share information with other databases, as Yaniv said before, with Phenome, and also to use tools that this database or server has. This is the image of the web page. In this web page, we have a login place here, and in the top part, there is a series of pull-down menus for to serve the ontology, the entering the experimental module, the GenPlasm, the phenotyping. You have also a bar with feature menus with tools, and in the middle there is a summary of the data stored in the database, such as the GenPlasm, experiments, variants. There is the map of the accessions or the biological status, some images, the type of trials stored, and some links to work that we have done, such as the survey of designing your ideal tomato. And in the here with a carousel with the main source and collaborators, and here information about us, help, and other things. So let me go to the Harnesstom database. Okay, do you see the gateway in the page? Okay, so as I told you, there is a drop-down menu in the Harnesstom. This is the main gateway. So here you can see the GenPlus summary and the mapping population. There is a summary of the data or metadata stored in the database, the map of accessions, the number of accessions in the map, some images that are random and change every day, the type of experiments, the link, and the help. So to access, for example, to the experimental module, to the program, we can create, upload from Excel or search. If you click here, you can see the information about the programs that are stored under the Traditom, and you can see the name, the abbreviation, the objective, the crop, the external reference. Here you have links to the main pages with programs, the contact leader. If you click here, you can see information about and the email of the main the leader of the program, and in details, you can see detailed information of the objective or other information related to the program. Appended to this program, there are some trials. Here is the list of the different trials we have. A trial is a compendium of one or several experiments. We have the abbreviation, the name, the description, also the program associated, the type of trial, the public release date. And if we click in details, for example, we can see all the description, the start date, and the public day about the trial. We can see also the studies. For the studies here, we have the 24 studies for the TomGem, and for the Traditom experiments, we have different descriptions about the trial and the experimental factor used to analyze these samples, the growth facility, the plot design. In the description, you can see more detailed description about the location, the growth facility. We are going to implement also more description about the study and the location details, and you can see all the related germplasm used in this study, with links to the institute code, detailed views of the germplasm, and the accession. So let's go to the germplasm. If we go to the germplasm, here there is the list of all germplasm used in a story in Harnesstom. The ID, the code that gives this germplasm for the project, and its maintainer number, and for which project is. So if we go to this, we can see accession details. We have the multi-prop passport descriptor with accession basic information about the collecting site or accession number, biological status, ML status, and the taxon with links to NCBI. The collecting site information for this accession, we don't have information. The maintaining institute and the institute that donate this accession, and some breeding info. Also the attributes that this accession has. And let's go to check another image here. In the top part of the page, you have the quick search, so you can see different. For this germplasm that is in the Traditom project, you can see more things. You can see images with description about the development states and anatomical entity, and which germplasm are related to this accession. In the germplasm module, we also have different things and germplasm per study images, but they don't show at this moment. The cross, we have information about the crosses stored. This is the cross that is used in TomGem, the parents, the study. We can have more details. We are planning to put here the germplasm related. We can have the pedigree. So these are the pedigrees stored in the database. If you can see more details, the germplasm related. The mapping population, if you click in details, you can see also the germplasm that are from this mapping population. In the mapping population, you have the name, the cross, and the generation if you want. And let's go to phenotyping. For phenotyping, we can have observation levels that are accessions germplasm in an experiment, and we can have the observation level, the block, the experimental design, or more details about the coordinates. We can store also information about samples that can be taken from an observation unit or directly from a germplasm. We can have more details about the developmental stage or the anatomical entity where they are taken. And then we can have the observation values. Sorry, it's not, okay, it's taking a little. Take a little, we will need to speed this, but there is a lot of data. Wait a minute. Okay, so we have the observation variable name, the observation level, and the value. We have information here about the observation variables, and in details, we can have information also about the scale and the method used to create this observation variable and the trait. In the genotyping module, we have the genotyping platforms with the sequencing type, the name, the description, the genome of reference, the marker count, the method, the methods to get this, the reference assembly link, and some of more detailed information about the method in the details for the markers. We have information about the name, the type, the chromosome, the position. If they are from PCR or other type of markers, we can have the start and the end, the reference allele, the platform. And in details, we have more information about, we can store information about the primers used to analyze this marker. In the variant catalog, or variant metadata, we have information about the variant sets that we have in this in Harnesstom. For example, we have the Sol cap or the Traditom variant set, and in details, we have information how this variant set is obtained, which filters are used, and you can download it from this link. At this moment, we don't have habilitated the variant set. Now we are working in preparing this view, but at this moment you can see the germplasm value for each genotype. In the variant catalog, we are working also in the view of this part, but you can see the different QTL studies with information about how they are done. In detail, you can have more information and also the studies that are related with this QTL study and the QTL variants. As I told you, we are working in that, so we need to put the phenotype, the observation variable on the chromosome, but you can see the name and all the info associated to this QTL. And also, if in details, we will see, because at this moment not, if there are the marker and the gene, if they are gene associated. The same for variants. We can see the GWAS variants, sorry, the study information about the QTL study, all the information that is about how this QTL study was done and the related studies. And in the GWAS variant, we can see the name of the variant and statistics about this variant, like the minimal frequency, the sample size, the p-value, the allelic effect, the study associated, and is prepared to store the output of different GWAS for different software, as the same as in the QTL variant. Okay, let's go. We have different kinds, a part of the quick search that we have in the upper part of the if page. We have an advanced search that allows to filter accessions by passport data. So we can, it's a multi-filter. Here appears the number of accessions, the distribution of accessions in each country. For example, if we want to search for Italian accessions, let me click. Okay, it filters the risk. We have around 450 accessions from Italy, but we want also that they will be advanced on improved cultivars. So the accession now is filtered. And we want that they are collected, they are from an institute. So we have one accession. The filter, as you see, is change the count, taking into account all the filters. You can remove also the filter. Another search tool that we have is the Gourmetom that is created for non-scientific users, and it allows to filter by country or biological status or by different traits for which we are designed some icons. In this case, the filter allows to filter by different accessions. We will have at this moment is only available Traditom data for this, and we can filter by different parameters. It is not working. Okay, so let's go to the features. We also create an API platform to exchange data and to have access for the data. So we have different functions. So we want, for example, to access to the information of one accession. We can put the ID of the accession in the database and execute. And for this node, maybe 1000, execute, 1700, sorry, because I don't remember the IDs. Okay, what I will get with that is the accession number and all the info associated for this. This will allow to connect to Phenome, to SGN, or to breed-based. Finally, we have also the graphical filtering that we have implemented from the breed-based page. So we can select a study. We can select the unit from we want to observe at plot level or plant level. We load the data. So here we have all the data that is in this experiment of TopGen with all the variables. And then we can apply the graphical filtering. So we can select a trait. We can put the flower number. Here we have the distribution for all the experiment. We can click and define the area that we want. We can add a new variable, and we can select if we want the logical combination. If we want that will be and or not, we can select that the plants have this range and the fruits and percentages that the list is automatically filtered. We also can do the same, but grouping by accession. Okay, here there is appeared the count. So for the control moneymaker in the experiments, we have 25 plants, 35 plots. So we can do the same. We can filter by different, sorry, if you can add more than one variable, you can click in the group, or where you can delete from here, and we can add a new range and select, for example, fruit number. And the graphics and the data is filtered. So let me go to the presentation. Okay, let me share my screen. Okay, so it's just the last things is that for Harnesstom version 1, the next steps is compilate and upload experimental data from a modern tomato database and projects. Actually, we are curating data from the Traditom core collection and from the Sauvage experiments from the INRA, and we need to habilitate the load tools, the QTL species module, and the allergen plus B, one publication reference links, and fix other things in the database, and also connect Harnesstom to Phenome, Gesol Genomics, and Tom Express. And when we have this connection, we can expand the visualization tools and for analysis, such as GBROS, GRAPS, breeding tools, or other statistical tools. We are also working in create a tool to request seeds to the gene banks, and for the new version, we want to adapt the database to include RNA-seq, structural variants, and GWAS and QTL meta-analysis. Okay, thanks to all the people that have collaborated, especially to Dave, that is the heart of the database, and Edan, that was working at the initial steps of the database building with me the templates, to Victor, that is working with to implement the GBROS and all genetic tools, and the people that has helped to develop the templates, Antonio Monforte, Maria Jose Diez, Matfield, Mara Ercolano, and Julien Bay. Okay, thanks all. And if you want to do any question, put in the chat.
Thank you, Clara. Thank you very much for this a very useful overview to start to use this database, I think, in the future. So maybe we have to learn how to use better in the next. In the next. I will just make a quick question because we have a little bit of time. What about the variants? I mean, the variants are reported to the genome, the last genome.
Mara, you have silence. I can listen the last.
Yeah, because there is a noise outside. Okay. The variants are reported to the last reference genome that has been released, or are mixed, so are related to the original experiment.
They are related to the experimental, to the original experimental genome version, but we want to do a conversion, or to try to put the synonym in other version of the genome. At this moment, they're just as they are reported in the original paper or in the engineer work. In the case of SolCap, as we have the 3 genome versions, it's easy to convert because we have the synonyms in all the versions of the genome. So when you find a variant, you can find it in the different genome versions, the marker and the position.
Okay, thank you. Dani is asking to know how we can get the EDA password.
Okay, the EDA password, you can go to the login and then register. And if you are registered, you have full access to the data or to some functionalities, to your own data. You can edit, upload your own data, share your private data with the people that you want to work, submitting things or editing the data, and that's all. If you go to register, you can register and access to more information. You can submit data also, sending us all the templates. We have prepared templates, and you can fill these templates and send us these templates in bulk with all information, and we will check that all is correct, and then we upload this data. So there are different things, but filling create forms one by one, and also in bulk.
Okay, thank you, Clara. So if there are other questions, please write on the chat, and then at the end of the session, we will discuss it. Now, let's move to the next speaker, that is Giovanni Giuliano.
Hi, everybody. I had a question. Can I ask it by voice?
Yes, please.
Two questions. One is, what is the division of labor between the Phenome database and the Traditome database? And second, how long is the Traditome database going to be maintained after the project end?
Okay, the Harnesstom and the Phenome, we will share most of data, but they have different goals. One is for researchers and for curated data. And the Phenome can also include that raw data, and is more focused to breeders and to companies. And how long this database we will transfer to Sol Genomics when the project is finished and the funding. Sol Genomics, we will host this database.
So there is no provision for a European database to host these data.
I don't know for the moment, but it will be in SGN.
It will be in SGN.
Giovanni, you know that this is difficult to answer now. There are no provisions because there is no money, there is no funding currently. We should work together, maybe the frame of grace infrastructure.
That's one of the next challenges. I mean, so
one step at a time. What a step at a time.
Yeah, sure. Okay, so thanks, Clara, and thanks, Mara, for introducing me, and thanks to the organizers for inviting me to give a talk on genotyping techniques. I hope you will resist the hypoglycemic moment before the lunch. And let me share my screen. Do you see my screen?
Yes, yes.
Good. So let me put it in view mode. So my title is Genotyping for the Management of Large Collections and the Reconstruction of Domestication History. I'm Giovanni Giuliano at Enea, Rome, and participate to Harnesstom, and we coordinate work package 4, which is the technology work package of Harnesstom. So it deals with several issues, including resequencing and genotyping. And I'm sorry, but I think somebody should mute their microphone. Mara, can you please mute?
I don't know how can maybe from the CMO computer is possible to shut down.
There is somebody with an Israeli name that has the microphone on.
Simo or Dave, you are under control of this session, please. Can you mute this or silence this? Is it working? Yeah.
Yes, okay.
Okay, now it's working. I was silenced myself for a moment. Okay, so please keep your microphones on mute. So let's go back to the presentation. So I will be talking mostly about results obtained in the previous project that dealt with the 4 major solanaceous crops, the G2B Sol project, where there was a lot of massive genotyping done in that project, as you will see, and we learned quite a bit of things about both management and of large collections and reconstruction of the domestication history. And we are now working actively, in spite of the fact that the project is finished, on the whole genome association part. So this is the project. It had 19 partners, some of which participate also in Harnesstom, and it was focused on the 4 main solanaceae: tomato, potato, pepper, and eggplant. And I will show you results from the 3 of them, tomato, pepper, and eggplant. So these are the accessions that we had in G2B Sol, about 65,000 accessions, of which 14,000 wild, distributed among several worldwide gene banks. You can see the project had partners all over the world. And tomato was about 25,000, potato 17,000, pepper about 14, 15,000, and eggplant about 7,000. And for the first, and what is this? This is, I like to compare it with the priceless phenotype library. This is the Biblioteca Angelica in Rome, which is a very precious collection of ancient books. However, the problem with this library is does not have a comprehensive catalog. So it's very difficult to find the book chapter you're looking for in this library. And that's the same with gene bank collections. There are passport data that are often not complete, classifications, and sometimes there are different systems used by different gene banks. So the first step was to collect all these passport data from all the gene banks, phenotypic data, and images. And there was a lot of manual curation done on this data. This was, for tomato, it was mainly done by Maria Jose Diaz group in University of Valencia. And as you can see, we have in total about 56,000 passport data, about 56 passport data for 56,000 accessions, phenotypic data for 28,000 accessions, images for 17,000 accessions. And they are, for tomato, for instance, we have 23,000 accessions with passport and 12,000 with phenotypic data and 10,000 with images. And all this is available at a one-stop shop, which is the magic keyword here, and you can access it. It's publicly available here at this address down here. Now, the second step, which is what we will be talking today, is use genotyping for sort of putting order and reconstructing the catalog of these accessions, of these collections. And here you have examples of both non-targeted genotyping methods. Increasingly, the non-targeted method of choice is whole genome resequencing. For tomato right now, you can resequence at 10, 20x with around 100 euros per sample. And you get millions of SNPs and structural variants, small structural variants. And for resequencing, you need a reference genome, of course. And then there is a second very used non-targeted method, which is genotyping by sequencing. And what you do there is you restrict your sample and you sequence, you make short reads flanking the restriction sites. So it's what's called reduced generation sequencing. The generation sequencing, and it does not need a reference genome. What's the drawback of this? It has a high percentage of missing data because very often, especially in interspecific populations that have more than one species, there is a loss of a cut site. So you are losing the sequences flanking this cut site. And you get thousands of SNPs and about 10 to 20 euro per sample. It depends on the combination of restriction sites you are using. You can go up to tens of thousands of SNPs. And then you have targeted genotypic methods. The most used up to a certain point where in Phenome DNA chips, in which you have one SNP per bead. They are bead-based, these chips. And the main problem is that they detect only diallelic SNPs because they use mostly two colors. It's not a big problem because anyhow, the bioinformatic pipelines that are used for downstream processing of these data are mostly created to handle diallelic SNP data. And then this is the most recent one. It's called single primer arrangement. And the idea is you have a single primer, you target regions in the genome. And instead of doing a bead that targets a single base where the SNP is, where the target SNP is, you just generate a single primer that you then use to sequence around the target SNP. So you get around 100 bases around the target SNP. And the bonus feature of this is next to the target SNP, you can discover many non-target SNPs. So it's a target method in which you target specific regions of the genome, specific SNPs, but then in these regions, you can discover new SNPs that are present in the genotypes you're using. And just to give you an example of how you can play around with these SNPs, the flexibility of SPET, this is a 5,000 SNP panel that was developed based using mainly intra-specific target SNPs. So SNPs that are present within Solanum melongena. And you can see if you use the target SNPs, you get a very good resolution of the melongena accessions, which are the purple ones. While all the wild species cluster in a very small cloud here, because the target SNPs are not very good at discriminating interspecific differences. On the other hand, if you use all SNPs in which the non-target ones prevail, they are about 2 or 3-fold more abundant than the target, you lose resolution of the melongena accessions. They cluster all here, but now the non-melongena are much better separated from each other. So depending on what you use, you can make intraspecific discrimination of very closely related accessions and interspecific, for instance, for classification of species. And another interesting bonus feature of the target ones is that if you classify these accessions by origin, this is the origin from the passport data, you can see that different geographical regions separate in this PCA analysis into different regions. So the target SNPs not only separate well melongena, but they separate accessions of different geographical origin. And this allows you to make a lot of reasonings around the domestication and also around the migration of the various, after domestication of the various populations. So we barcoded, the step 2 was using these different technologies to barcode about 40,000 accessions of the 4 crops, about 10,000 of tomato, 5,000 of potato, 10,000 of pepper, 16,000 of tomato, 5,000 of potato, and 3,600 of eggplant, which means about 70% of the active accessions that were present in the project. And this was a very big effort, and it worked amazingly well. And here you get a little bit of comparison of the efficiency of the various methods, because for pepper and potato, we used GBS, and we used a mean of about 1 million Illumina reads per individual. And we got for pepper about 3,000 high-quality SNPs. For potato, which is much more polymorphic, is tetraploid, 13,000. With SPET, which is a targeted method, it's much more efficient. So on average, you get a much more high quality SNPs per read than you get with non-targeted methods like GBS. That's the one difference of the two methods, target and non-target. So what do you use these SNPs for? As I told you, the target ones are very good for discriminating closely related accessions, intraspecific. Again, here is from eggplant. What do you do here? You take replicates of the same genotype. This is the reference genotype of eggplant that you sequence 67-3/3, and you can see that the vast majority of these replicates are in this region here with a dissimilarity pairwise difference of about 0. And then there are some that go up to 0.003. So what you do here is you put a cutoff and you say, okay, above this value, I can consider this. Everything that falls in this is the training population, let's say so. So it's made of genetically identical individuals. Now you go to your test population and you genotype them and you say, everything that falls on the left of this line, I consider duplicates. Everything that falls on the right, I consider different accessions. And these are the groups that you find in this very large collection of about 3,500. You find the groups that are of up to 12 different individuals, etc., etc. The vast majority of the groups is made of 2 individuals. This is an example of different accessions from different gene banks. You can see that have been classified as duplicates. You can see that, in fact, they are very similar to each other. And you can score what we call auto-duplicates, which is the percent redundancy duplication within a gene bank. This is CGN in Wageningen, this is in Rye, this is the World Vegetable Center. And you can also score alloduplicates between different gene banks. Okay? Now, the second thing you can do is reclassification. So very often the classification of species is done by gene bank curators that are not very familiar with all the species within a species complex. So sometimes they make mistakes. And here is the reclassification we did for the eggplant collection. And you can see here we use the non-target SNPs that I said before. They discriminate better between different species. And you can see there is some change in the number of accessions in different species. It's not big, so it means the gene bank curators did their job right, but there is some difference. Third use you can make of these techniques, again, I'm referring to SPED because most of our data with SPED is to use both target and non-target SNPs for the fine mapping of recombinant inbred populations. And here you have the BIL population that I think was mentioned by Shai Torgeman in this morning talk. And basically, this is these are recurrent parents of Solanum lycopersicum. This is the wild accession. And here, given that you have an interspecific cross, you can use both target, but probably the most discriminating ones between pennellii and lycopersicum are the non-target ones. But then, if you want to score heterozygosity within the BILs, then you can go to the target ones. Again, it's a very flexible approach that allows you to either score alien integrations from faraway species like Solanum pennellii, or you can score using other SNPs. You can score recombinations between two closely related parentals of tomato, like you do in breeding programs. Most breeding programs are between closely related accessions. They are not interspecific. And this you can use then to map putative recombination hotspots on the genome. So you map genetic distance against physical distance, and you can see that there are recombination, what you think are recombination hotspots in the genome. Now let's switch to last part of my talk, which is domestication and diversification. So Darwin noticed that when you domesticate a species, you start with a relatively invariant phenotype in the wild ancestor, Canis lupus in this case. And then when you domesticate, you are stricken by the vast diversity in shapes. This is a collection of different dog races. You can see how different they are from each other. And this is a collection of different tomato genotypes, and you can see how different they are from each other and from their parent, Solanum pennellifolium. So how does this happen? This happens essentially through this is a mapping of the morphological diversity of the wild ancestors of tomato. The first serasiformes, the first wild serasiformes, you see there is a little bit of increase of morphological diversity. Then the serasiformes vintage, these are already domesticated accessions. You see there is a tremendous increase of morphological diversity. There is a decrease here, but then it keeps being high. Maybe you have a decrease in morphological diversity in modern varieties due to a selection for long shelf life that was done recently by breeders that also entailed a decrease in the morphological diversity of tomato. But this is very different from the genetic diversity. You can see here there is very high initially, then it goes down, then it goes up again. But then in the vintage, it goes down to very low genetic diversity overall in the genome. And then it only increases in modern varieties. And this is due to the introgression breeding done in the 20th century, 20th and 21st century. So you have two opposite trends, morphological diversity and genetic diversity. And the main drivers of the decrease in genetic diversity are genetic bottlenecks that are genome-wide, and of course then genomic sweeps at selected loci, in the sense that of all the possible alleles that you have at a certain locus, the breeders select few that give the desired phenotype. And again, even at selected loci, you see a decrease of genetic diversity, but an increase of morphological one. And this is the difference through the years of morphological diversity in the various groups. And you can see as the time goes, the morphological diversity increases, as we said before, and then it plateaus around 1800. Okay. And but another take-home message is that many of the different shapes that we have in modern tomatoes, that the kidney shape, flat, cylindrical, pear-shaped, that you find in very early South American cerasiformes, early domesticates. So the idea that the early cerasiformes were small tomatoes resembling pimpinellifolium is basically wrong. The domestication that occurred in South America already selected early on for different shapes and sizes. And then finally, you can, having georeferenced accessions and drawing similarity matrices between them, you can attempt a reconstruction of the domestication centers that are known. In the case of pepper, for instance, it is known that it's Mesoamerica, and of the migrations, of the subsequent migrations. And for instance, for pepper, you have the domestication center, then you have an expansion in South America, and then from Brazil, you have a route that goes all around Africa, which we call the Vasco da Gama route. And it was probably made by Portuguese traders. It goes all around Africa and arrives to the Far East. And that's how pepper arrived to the Far East. And there is a second route that goes directly from North America, directly to the Philippines and then to Far East. And these were the galleons that went from the Spanish, and this was a trans-Pacific route, from the Spanish colonies in Western Mexico, all the way to the Philippines that were also Spanish dominion at that time. And this is eggplant. And here we had very good evidence for two independent domestication centers, one in Southeast Asia that is more closely related to the Chinese and to the Japanese accessions, and then an Indian one that then gave rise to the Middle Eastern and Southern European ones. And this route here was probably mediated by Arab traders. There are some paleobotanical evidences in Jerusalem that suggest that it was the Abbasids, the Arabs, that brought eggplant to the Middle East first and then to South Europe. All this is published, and these are the papers that you can go and see. And finally, you can use historical data, gene bank data. Of course, you can use also data that you collect in field experiments. But for the moment, I'm going to focus just on the historical data, gene bank data. And you can use those data in combination with genotyping data to do GWAS. And you can see signals. These are signals for fruit shape and size in tomato. You can see around 25 genomic regions that show selection, of which 16 have no known candidate gene. They are new. So even from gene bank data, you can get new possible QDNs for fruit size and shape. And the interesting thing is that given that these are georeferenced accessions and more or less it's also known when they were generated, you can see when these different genetic sweeps were done. So this is, for instance, the pink ones are genetic sweeps that are uniform in all cultivated accessions. So they were probably very early. These are wild alleles. The purple ones were selected more, these one here, were selected in modern accessions, etc., etc. These are alleles that are very rare in Italian accessions that are, in our analysis, clearly separated from the Spanish ones, and so on and so on. And the last slide that I wanted to show you is that you can do this for different species. And as you know, tomato and eggplant are highly synthetic. This is a synteny map of the 12 chromosomes of tomato here and eggplant here. And you can see the bigger chromosomes are interestingly more synthetic than the smaller ones that show higher translocation rates. And this is a recurrent phenomenon in the Solanaceae. We have to find an explanation for that. And you can see this is a QDL that is synthetic between the two species. Another region here, highly synthetic. Another here, synthetic. So you can find synthetic regions. It tells you that probably it's the same genes that that control size and shape in the two species. And here, that's a nice one. You have a translocation between chromosome 11 and 12 in the two species. And look, you have a region here that is on chromosome 12 in eggplant and chromosome 11 in tomatoes. So you can find, of course, this is the beginning of a very complex analysis, but it's very interesting that you can use historical gene bank data and genotyping data to find novel QDLs for size and shape, and for many other traits that gene banks score during regeneration of the seeds. And with this, I finished, and thank you very much for your attention. Before I close, I want to make a advertisement. We have another training school in a few days. We have an international workshop and training school on the sustainable management of plant genetic resources. It will be held in presence at the Mediterranean Agronomic Institute of Chania. But if you are interested in participating online, the participation is completely free. Like for this school, you can find the program and the free registration online at this address here. And if you are not able to find, please write to me and I will send you the link to the program and to the registration site. And thank you very much for your attention.
Thank you, Giovanni. Thank you to give us this overview about NGS-based marker and on their use for several possibilities. So let's see if there are any questions for Giovanni, for other speakers.
Yeah, I can see one from Rafael Fernandez Munoz.
Right. Yeah, the first one is from Rafael.
Yes, so how is about using SPETs for biparental populations? That's a very interesting question. So we just did a new 12K SPET panel. And we asked a question, the following question. With this, we genotype different collections, one from UPV in the frame of Harnesstom, one broad one from UPV, one narrower one, which was mainly land races from University of Viterbo, and one from Naples, Mara.
Yes.
That was, I believe, a biparental population, correct?
Correct. Exactly.
Okay, and that's interesting. The biparental is the one that in the target SNPs shows the least diversity. So out of 12,000 SNPs, target SNPs used in this panel, only 2,000 to 3,000 are polymorphic in your biparental population, while the highest level of polymorphism, around 80%, is in the UPV population, which probably has a very broad genetic basis.
Right.
But the beauty of SPET, I'm not getting any money out of the selling of SPET, so don't misunderstand me. I'm just trying to depict the advantages. is that if you go to the non-target ones, you find plenty of SNPs that are very good at discriminating your narrow basis by parental population. So again, by using different classes of SNPs, you can even discriminate between very closely related, with parental populations between very closely related accessions. So yes, the answer is yes. SPED is very flexible and allows to do this.
Yeah, sure. So any other question for Giovanni or for the other speakers?
Well, and to answer Rafael, in that case, we use non-target SNPs that are discriminating in that particular population. So the general rule that I expressed before, that non-target discriminate better interspecific, is not general. If you select your non-target ones, you can find ones that are very good at discriminating between closely related accessions.
Well, if there are no other questions, I was just very interested to the slide that showed the sweeps that you found in tomato, the one that there are 25. Most of them you say that are in there are loci that are unknown loci, right?
Yes, this was GWAS done by Jose Blanca using the 5,000 region panel, SPED panel, and the historical gene bank data from around, I believe, 10,000 accessions with size and shape data. So it's a very large experiment.
Yeah.
Okay. And you find many, many regions. And the interesting thing is, as I repeat myself, is that you can even say approximately when and which subpopulation the sweep was generated. Was it generated by modern breeding or by the ancient Amerindian populations? You can relate that. And the fact that it is synthetic with eggplant, it's very reassuring. So it tells you that there are probably common mechanisms. You expect it to be synthetic because you know these genes that control size and shape are very conserved, even among non-Solanaceae species, as was shown by, for instance, for the ovate family.
Yeah, yeah. So we will read the final result in your published paper.
Yeah, we are writing it up right now.
Okay, thank you, Giovanni, the other speaker. And so we can move to the final discussion with the different partners that are involved in it. So we can try to have Toni, Yaniv, Giovanni is here, Dani Zamir, myself, Toni Monforte, and Christian Beckham for the final discussion on this topic. Toni?
Okay, I can start, but I think that...
Of course, you have to start, you are the coordinator.
No, I think the future looks very good for all scientists, not only because we are able now to screen large populations for phenotype using new techniques and new machines, and we have lots of accession to historical data, to passport data, to pictures and genotypes and sequences. Also because something that we don't really have highlighted here, but some partners did, final confirmation by genome editing is now in everybody's lab. So if you have, and I think this is for the first time in the history, we have this possibility not only to obtain a lot of genotypic and phenotypic information, but also we have a tool that quite quickly can you can test the hypothesis of the involvement of the gene in the phenotype. Okay, but maybe.
Thank you very much, Toni, to give us this outlook. Yaniv, you want to say something?
Yeah, I see that Bachem is not with us.
Okay.
Anyway, yeah. So there are a few issues that we should consider for the future. One issue is SGN. As you know, SGN is not really maintaining the data since they are not funded to do tomato. So basically, I'd say that nothing really happened on SGN in the past maybe 8 years or more. They do a lot of other stuff that relate to the project they are funded to do. But as far as tomato, there's nothing done. So genes that were published are not updated. If you search, they are. This is a tool that is important, such a tool that will be available for the community. And this is something that we have to consider as maybe as part of the, the database, the beautiful database that was created for Harnesstom. I haven't, just by the presentation, it looks very impressive, but if you put it in SGN, nobody goes in there for the tomato because people realize that it's better to go to other databases for tomato. So we have to this is why I wanted Christian to be in and to see how we can create something that will serve the community, all aspects of the data. This is a future. This is something that is very important for the future of the community. So this is one thing that I wanted to raise. The other thing is the TGRC. As you know, I'm a great admirer of TGRC, and I know it's tremendously important for the community and the tomato work. Charlie Rick is up there, so he's got all the information and everything, but he can't help us. Roger is very healthy and looks great. I saw him last week, but he's retiring. And there is no, Roger is a scientist, so he has excellent publications and so on. There is no plan for the continuation of the TGRC. So the technicians that work there will continue to send seed, but we need, the community need the scientists that will investigate these resources and do up-to-date science and improve the quality of the TGRC collection. So one of the things that I told Roger, apparently that now they don't plan to replace Roger with somebody who will be responsible for TGRC. They will ask people around to take, kind of. general responsibility, but not the scientific responsibility that Roger and Charlie had. So the dean doesn't want to do it. So I said, okay, let's send Dean Davis 1,000 letters from Solanese scientists and request that, urge him to how important it is, how key it is for the future of the tomato work. So again, Bachem is not here. I will catch him later. And if you have any idea, as I'm retired, and if you have any ideas that could promote these two things, one is to have a bioinformatic engine that could really serve the young people in the community, and the other is to help with the TGRC. And of course, this requires to write new grants to fund some of these projects. So I can see if we write, if you write an EU grant that includes as one of the components, kind of a continuation of Harnesstom, that it will include as one of the components a local, some tomato genomic database. That's one solution that I can see, and I'm sure that if we can, once this is done, then companies would be willing to chip in and raise some initial funding for this type of work.
Okay, thank you. Thank you very much, Dani, for this point that you raised. And maybe we can say, ask Giovanni if he has some it for the future.
Yeah, so I would like to answer both the point that was raised by Toni and the 2 points that were raised by Dani. Toni said, okay, we have all these QDLs and traits, and we have genome editing. And so we are in the position of testing all these genes by high throughput genome editing. It's not so simple. So the problem is that biparental populations like the ones that Dani created give a very good resolution, but they don't encompass all the variability of a species. Only the traits that are contrasting between the two parentals. And the second is that large populations like the ones that were phenotyped in Traditom and Harnesstom and Giuliano have a very high level of linkage disequilibrium. So the QDLs that you find there comprise hundreds of genes. So if you have like for tomato, we are finding the GWAS on the Giuliano core collection, hundreds of QDLs. Each of them has around 100 genes. So the main problem is, how do you select the genes to then test functionally by genome editing or by various virus-induced gene silencing or some other functional method? So I think that there is a missing link in the fact that, repeat, biparental populations only select. They don't select all the possible loci that control the trait, but the ones that are contrasting between the two parents. The large collections, on the other hand, have very large QTL. So it's difficult to focus, to zoom in on a gene, as you do with biparental populations. So we have to find creative ways to combine the two. And I think that one thing that we have to think about, and is worked on in Harnesstom, is try to find a way to increase the combination rates. That will be key to zooming in easier on single genes rather than on very large QTLs. Regarding the 2 points that were raised by Dani, as you know, I'm coordinating this project on the building of a future research infrastructure on plant genetic resources. And from the type of calls that are going to be there, I think that this future research infrastructure can provide hosting for collections that are becoming orphan, like TGRC. There would be lots of the European gene banks that would be interested in hosting a copy of TGRC accessions. Like Huji, I mean, sooner or later, your fantastic collection at the Hebrew University will have to be hosted somewhere else as well. I mean, I see it much more difficult to find money for doing immortalizing the bioinformatic data. This is not that money, in my view, will come from specific calls in Horizon Europe or other projects. So we have to lobby hard for the bioinformatics. It's a key point, and we have to convince the EU that we have all this incredible amount of data, and we risk them getting dispersed. So an investment in bioinformatics is essential for maintaining this data that we're collected through different generations of projects. Of course, we can connect with the Elixir research infrastructure, but I doubt that they will make this their main goal. They are dealing with so many different things. So we have to find money in Horizon Europe for this.
Okay.
Just a comment. I was not so simple thinking, like,
of course
you could go from QTL and GWAS analysis down to the gene and prove it, but I was just indicating that now is a very good moment because we have for the first time so much data available and also techniques that can give us responses once we identified good candidates. And for this, there are lots of techniques and approaches you can take to get out to a more precise candidate. And for the other, all the gene banks know that this is the nightmare that they go through because they have permanent funding for these long-term requirements or needs. Is what is going on in all the countries that have germplasm, and the funding agencies in Europe go through projects, and projects have a span of period. I mean, they are 2020, 2030, 2000. So there is a limited amount of time in which you can get the funding, and then you need to renew this. So the best thing we can do is just that the calls has this topic about genetic resources and maintaining the genetic resources, both information and the stocks of seeds alive and available for everybody through different calls or through different periods of calls. And this is, I don't know if there are previous cases of such a thing, not just on genetic resources, but in anything, or just go by, as you know, there has been in the Horizon, there has been a lot of diversity calls. That now maybe there is no such a thing in the new call. So it goes with like in waves.
Yeah, I mean, the EU is very simple-minded. it thinks, okay, we do the diversity call for a couple of framework programs, so the diversity problem is solved in their minds. Of course, not in ours. And then they move to something else. That's the way the reason. There was this microalgae boom, and then they thought everything in biofuels was solved, and nothing was in reality solved. But it's the way the EU reasons. So this attempt to generate a research infrastructure is to commit not only the EU, but also national governments on a long-term program to have a network of gene banks all over Europe that collaborate, work with similar standards, and you can have also a optimization of resource and use if you have this large network.
Yeah, I agree with you. The problem is that we cannot wait for the agency, the European agency, to take care of the collection that are spread already in Europe, but now we have this big problem for the collection of US. This is a very big problem because it's a very good collection. So maybe we have to try to wait to contact somebody that is in a different field, like in the national agency or let's say politics, but I'm not sure that is the right way to go, because we cannot talk only as a scientist. We have to talk as a society. This is the problem now for the conservation of germplasm. And of course, the conservation of the germplasm is the first point that we have to look for. But the other problem is also the conservation of the information, because I mean, it's a lot of work making this database. We cannot consent that this database will be lost in the future, because we invest a lot in that. So we have to try to maintain such a platform for all of us. And of course, I say, Toni, this is a good moment because the technologies is going fast and we have several options to solve our problem. But we have to have a good infrastructure to use them. So it will be a little difficult in the next few years, let's say 4 or 5 years, to at least start to build up something for the future. And I see there is a Carmen Leda that say a crowdfunding project is something in real. What do you think about make a crowdfunding project for the genetic resource? And
if you convince big industries like Shell, Syngenta, these ones to become the crowd that funds, then that's feasible. But if we just ask individuals to crowd fund all this effort, I think it's unrealistic. So we could convince, big industries have lots of money to spend for tax breaks, and they need to do, many of them need to do some greenwashing, isn't it? So I think there you have good chances of getting good contributions if you clarify what the money is going to, because they like to see the money being used for something precise. I think individuals, I don't see it feasible. It's in Europe only, you have 400 institutes dealing with plant genetic resources. How are you going to crowdfund 400 institutes?
well, anyway, you know, it's we can't give an answer now, but maybe we can decide that the only organization that could move such a project is the sole co-chairs. If we can encourage them to think along the lines that we talk about, and maybe arrange a meeting somewhere, or virtual or real meeting with some ideas. I think that if this is if I can convey this message to Bachem, who is the chair of the Soul Project, I think. Is he? He is, huh?
Well, I think that is
yeah. Maybe he's tired. Maybe he needs to be changed. I will talk to him. I'll talk to him and see. But this way, you know, the Soul Hoopla was a good way to fundraise, and we were successful in the past. So I think that we have to count on such an organization that will include Soul partners from all their countries, and if people get enthusiastic about it, it will happen. If not, then well, you know, we'll let things go. Maybe if you can talk to Bachem, also Giovanni.
Also Aureliano. At the moment, Ercolano should be the chair, if I remember correctly, because I should be co-chair, but I never get news on that.
Yeah, so maybe to revive it, or alternative, you know, to get an alternative group. Anyway, it needs to be done if we want to serve the community.
I think for the Soul, we need two things. One is to clarify who's the chair and who are the co-chairs. That's an important step towards and possibly elect them at the Soul meetings. Maybe the Canada one won't be the best because it will not be very highly attended, but we could have a system of national delegates, something like that, elected by national communities. And the second thing is maybe we want to make it more stable, like getting a tax code so that we can get contributions from companies, from privates, and use them for furthering the Soul adventure. This was a very nice initial effort that was based on volunteering for many, many years, but maybe at this point we would need a more organized thing, like a foundation or something like that.
I just wanted to add that indeed Aureliano is the chair, and it was decided, I think, in Greece. And but I think his initial job was to organize this, to start organizing the next meetings. So maybe there is a possibility that we propose that there is a new mission for the Soul. We started by sequencing, and then maybe sequencing more. Next step that maybe didn't made it so far, but maybe the new mission is about how the 2 points that were raised by Dani about how we maintain the resources on both information and physical resources, or any other thing. We need to put together, to get together and think about like a new Soul board that maybe could be able to, because all this about financing and having their own funding from private company was explored in the past by Christian, and apparently it was not so easy. even from the major investor at the time, that was Philip Morris, and probably now wouldn't be so much interested. But so
I think Philip Morris is probably gone, because I just learned that Nikolai Ivanov is not anymore at Philip Morris. He goes to the university. So
maybe where is the next? There is a meeting in Canada. Where is the next one?
The next one will be in Japan, and we are
next one is what?
Japan. Hiroshi is organizing. Ezura.
In Japan. Okay.
And in 25, we are thinking of proposing in Italy, maybe Ischia.
Ischia, yeah.
Maybe Ischia.
But, yeah. Okay, so maybe for Japan, something could be prepared, because not for Canada, it's too short. And it's not urgent for tomorrow morning, but it's urgent that we start thinking about it and make a plan. So here we are 4 already. You know, there are few people that can be recruited. Zach has to be there, Liebman. Some other people in US that do, you know, we have to get the younger generation also involved. So let's think about it. And since we are 4 here, I see 4 is Toni Monforte is here.
Yes, next to me.
Same.
Toni and I are together.
You're together. Okay, so we are 4 here. Let's think about it, correspond a little bit, and come up with, I'll think about it too. If I have ideas, I will write to you.
So let's go for a Sol 2.0, 4.0, okay? A new version that some joint project or global things to do together rather than individually.
Yeah.
Okay.
Right.
Okay.
This is a good mission, so we are taking an important task to do.
Okay, if there is no more question, no more challenges to embark, and I think it's time for going for lunch.
Lunch. Okay. Okay.
Thank you, everybody, for staying until the very last minute, and we hope that this course or series of talks about resources and the use of the resources to find out how plants do things that breeders and people are interested in case of tomatoes. Thank you a lot. See you next time.
Thank you, Toni. Thank you both, Toni.
Bye.
Bye.
Ciao, Giovanni, ciao.
Ciao. Ci vediamo domani pomeriggio.